Hello and good morning, and welcome to this class of Science and Technology, where we're going to continue with biotech and the applications of biotech. I hope you can all see and hear me. If you can write see and hear me, please write yes in the chat. All right, good to hear that. Okay, so I saw in your chat there were some doubts about current affairs. अभी मैं chat बंद कर रहा हूँ, उसको समझते हैं पहले current affairs को लेके आपके जो questions हैं, and then we'll discuss that. देखो, आप में से जितने लोग प्रीलिम्स रिविजन प्रोग्राम में हैं, you might also be reading current affairs with Mudit sir, right? अब देखो मेरी आपको क्या recommendation है, again of course you can choose to do whichever of these that you want. मेरी आपको recommendation है that the subjects of economy, science and technology and environment, इन तीनों के करंट अफेयर्स आप मेरे पे छोड़ दो और मेरे से पढ़ लो, एंड यू कैन स्किप दीज क्लासेस हियर. ये वाली क्लासेस आप स्किप कर सकते हो. क्यों बोल रहा हूं मैं ये? Mudit sir's content is probably the best current affairs content in the market without doubt. I think ऐसा content current affairs का है ही नहीं मार्केट में. किसी ने खुद न्यूज़पेपर पढ़ के, स्पेशली उनके कैलिबर के आदमी ने कहीं पे न्यूज़पेपर पढ़ के खुद पूरे नोट्स खुद बनाए हो, ऐसा आपको कोई मिलेगा ही नहीं. क्लियर्ड द एग्जाम थ्राइस एंड मेड रीड्स द न्यूज़पेपर एवरी डे फॉर 2 hours एंड क्रिएट्स नोट्स हिमसेल्फ. कंटेंट आपको मिल नहीं सकता. मैं भी नहीं बना सकता ऐसा कंटेंट, तो मैंने उनका कंटेंट पढ़ लिया. ठीक है, तो इट इज जस्ट मी रीडिंग द सेम कंटेंट एंड टीचिंग यू द सेम कंटेंट. द कंटेंट स्टेज द सेम, राइट? नोट्स में कोई फर्क नहीं आएगा, वही नोट्स हैं. बिकॉज व्हाई शुड बी डुप्लीकेट द एफर्ट, राइट? अगर मुदत सर कर रहे हैं, आई कैन से विद कॉन्फिडेंस दैट अगर न्यूज़पेपर से बताना हो कि क्या किस चीज पे क्वेश्चन आएगा और किस चीज पे नहीं आएगा, मुदत सर विल डू अ बेटर जॉब देन आई विल डू. बिकॉज ऑब्वियसली यार 3 बार एग्जाम निकाला है, ही हैज दैट credibility. But I would want to teach these subjects to you from that content, right? What static backup I have given you. तो स्टैटिक से लिंक, अब मुझे सर को एग्जैक्टली नहीं पता कि मैंने कहाँ पे और कैसे पढ़ाया है, राइट? सो फॉर हिम टू लिंक इट टू स्टैटिक इज डिफिकल्ट, फॉर मी इट्स इजियर. तो इन 3 सब्जेक्ट्स पे आप मेरे ऊपर छोड़ दो, मैं आपको करा दूंगा. बाकी आप करंट अफेयर्स मुझे सर वाले सारे देख लेना, ठीक है? इन 3 सब्जेक्ट्स के चाहे आप एक भी क्लास ना देखो, whatever he has written in his notes I will teach anyway. ऐसा हो ही नहीं सकता कि उन्होंने अपने नोट्स में लिखा और मैंने ना पढ़ा हो, because A, we make these notes together, we talk about it, we discuss it every day, and B, I am going to teach exactly from that content that has been made. That content is actually made a lot of times with the two of us discussing it, right? So I'm going to teach you everything that's in that content. तो आपको उसकी टेंशन लेने की जरूरत नहीं है. बाकी सब्जेक्ट्स का आप मुझे सर का देखो, इन 3 सब्जेक्ट्स का आप छोड़ दो, ठीक है? Is this clear? If this is clear and you have revised yesterday's class, please write ready in the chat. And today we will continue with applications of biotech. अब हम real life applications of जो principles हमने अभी तक देखे हैं, उन सब की real life applications हम पढ़ने वाले हैं, right? Please write ready in the chat if this is understood. Then we'll start. Okay, perfect. चलिए start करते हैं. Yeah. All right, let's start. देखो, बात करते हैं सबसे पहले ऑफ समथिंग कॉल्ड ट्रेडिशनल हाइब्रिडाइजेशन. हम क्या पढ़ रहे थे? हम कल पढ़ रहे थे बायोटेक्नोलॉजी. जस्ट गिव मी 1 सेकंड. सॉरी, कल हम पढ़ रहे थे बायोटेक्नोलॉजी. नाउ इन बायोटेक्नोलॉजी द गोल ऑफ बायोटेक्नोलॉजी इज यूजिंग बायोलॉजी टू बेनिफिट ह्यूमंस. यही गोल है. If I have to use biology to benefit humans, what do I need to do? I need to get better genes or beneficial genes. Let's say I'm a farmer who wants to grow certain crops. If I am able to use biology to get better genes in my crops, I would have used biotechnology, right? If I am, let's say, a cattle herder and I am able to get better genes into my cattle, which then produce more milk, I would have used biotechnology to benefit myself. So biotechnology is nothing but using these techniques, जो हमने देखा था, gene editing, principle of continuity of DNA, the universalization of the genetic code, right? ये सारी चीजें देख के अब हम देखेंगे कि इसको यूज कैसे किया जाता है, right? Now before all of this technology was known, उससे पहले भी तो लोग biotechnology यूज कर रहे थे. So before the technology was known, before anybody knew what biology is, how it's to be used, उससे पहले भी तो ये चीजें हो रही थी। तो उससे पहले ये चीजें कैसे हो रही थी? दे वर हैपनिंग थ्रू अ टेक्निक कॉल्ड ट्रेडिशनल हाइब्रिडाइजेशन। अब ट्रेडिशनल हाइब्रिडाइजेशन क्या है? देखो, 1 सेकेंड। या व्हाट इज ट्रेडिशनल हाइब्रिडाइजेशन? लेट्स से कि आपके पास एक क्रॉप है, यू हैव अ क्रॉप ऑफ लेट्स से अ व्हीट प्लांट। आपके पास एक व्हीट का क्रॉप है, राइट? अब व्हीट के क्रॉप के अच्छे कैरेक्टरिस्टिक्स क्या हैं? व्हाट आर द गुड कैरेक्टरिस्टिक्स ऑफ दिस व्हीट क्रॉप? गुड कैरेक्टरिस्टिक्स इन द केस ऑफ व्हीट क्रॉप वुड बी लेट्स से गुड यील्ड एंड लेट्स से पेस्ट रेजिस्टेंस। अभी बायोटेक्नोलॉजी, डीएनए वगैरह किसी को पता नहीं है, but obviously we still know that a wheat plant that has good yield and is resistant to pests is better. तो पहले हम एक टेक्निक यूज करते थे which was called traditional hybridization. Traditional hybridization. Now traditional hybridization has a certain understanding behind it. अब यहां पर क्या किया आपने? You take parent one, any wheat crop you take, let's say this, I will call this W1. This wheat crop had good yield, ठीक है, इसकी yield अच्छी थी. This is W2, इसकी पेस्ट रेजिस्टेंस थी. ये देखा गया था नेचर में कि ये वाला जो प्लांट है ये पेस्ट रेजिस्टेंट है, ये वाला जो प्लांट है पर्टिकुलर प्लांट, दिस हैज गुड यील्ड. अब ये क्यों हुआ होगा? व्हाई वुड दिस प्लांट हैव गुड यील्ड? Obviously it would have some good genes in its genome. Now I'm sure you are understanding these words. This W1 plant must have some good genes which were giving it good yield. This W2 plant must have some good genes that were giving it pest resistance. ठीक है, अब हमने क्या किया इन ट्रेडिशनल हाइब्रिडाइजेशन? अगेन वर्ड ब्रेकडाउन. ट्रेडिशनल बिकॉज इट्स नॉट यूजिंग जेनेटिक एडिटिंग और एनीथिंग लाइक दैट, वो कुछ हम नहीं कर रहे, तो ट्रेडिशनल टेक्निक है. सेकेंड, देयर इज हाइब्रिडाइजेशन. हाइब्रिडाइजेशन इज मिक्सिंग, ज्वाइनिंग. So we joined these two together and we, let's say, consider this the male and this the female, and then we try to have a offspring or a progeny, basically a child of these two. Let's call this W3, and we hoped, right, we tried that यहाँ की good yield से, इसमें W3 में यहाँ से good yield से characters आ जाएं और इधर से pest resistant के characters आ जाएं, तो एक ऐसा W3 बन जाए which has both good yield and also pest resistance, right? हमने एक इधर लिया जो गुड यील्ड का प्लांट था, एक इधर लिया जो पेस्ट रेजिस्टेंट का प्लांट था, दोनों को आपस में ब्रीड करा दिया. वी मेड देम हैव reproduction with each other and created a progeny. This progeny we assumed would have good yield and would have good pest resistance. Traditional hybridization तो clear है. Tell me what is the problem with this method? इसमें issues क्या हैं? What are the issues with this method? How इसमें कुछ प्रॉब्लम लग रही है क्या? वेरी गुड, सही बता रहे हो. गुड गुड गुड. हाँ, देयर इज नो कॉन्फर्मेशन दैट गुड जीन्स वुड बी ट्रांसफर्ड. देयर इज नो कॉन्फर्मेशन दैट गुड जीन्स वुड बी ट्रांसफर्ड. मेरे को कैसे पता कि यहां से जो ये गुड यील्ड वाली जीन है ये आ जाएगी, और मुझे कैसे पता कि यहां से पेस्ट रेजिस्टेंट वाली जीन आ जाएगी? अब इसमें भी, अब ये जो W3 है, W3 will also have certain traits for yield and pest resistant, but हो सकता है ना that W3 takes its characters of yield. अब yield से रिलेटेड जो जीन्स हैं वो यहां वाली आ जाए, और पेस्ट रेजिस्टेंट से रिलेटेड जो जीन्स हैं वो ये वाली आ जाए. ये भी हो सकता है. तो it gets pest resistance from W1 and it gets yield from W2. This is also possible. Then we have read and you have seen a video hopefully of this phenomena of crossing over also. So again, there is no guarantee. There is no guarantee that these genes would be transferred. ठीक है? अब we have to understand this in a bit more detail. I would not have taught this to you last year, but this year it's been in the news. There is Mendelian laws are in the news. So Gregor Johann Mendel, there was a scientist, आपने सुना होगा, एंथ्रोपोलॉजी वालों को बहुत अच्छे से पता होगा. There is a scientist by the name Gregor Johann Mendel who then understood these laws of genetics, and he is known as the father of genetics because it's his laws that we are all studying this from. बट आज हम ये नहीं पढ़ेंगे। और इफ वी आई जस्ट टेल यू द बेसिक्स ऑफ दिस, देख लेते हैं इफ यू अंडरस्टैंड इट। ठीक है, एक बार अगेन यू कैन गेट द नोटबुक आउट, एक बार इसके बेसिक्स समझ लेते हैं, राइट, हाउ दीज जीन्स आर बीइंग ट्रांसफर्ड बिकॉज दीज आर इन द न्यूज, ऑल्दो वी विल लुक एट इट इन अ लॉट मोर डिटेल टुमारो। ठीक है, बिकॉज अब मैं वैसे नहीं पढ़ाता, ये बहुत एडवांस चीज है, बट इस बार हिंदू में, लाइक आई एम टेलिंग यू अगेन, आई एम रीडिंग ऑल ऑफ मुदित सर्स कंटेंट, मुदित सर makes that content from the Hindu. There are 3 articles where Mendel's laws have been mentioned because of discovery of something called the jackpot effect in cancer cells. वो मैं आपको पढ़ाऊंगा कल या परसों में, but let us understand the basics of these laws. We'll understand the detail of these laws tomorrow. But basically वो क्या कहते हैं, अब देखो, let's say इसी केस को देखते हैं, right? Let's look at this exact case of one plant and having 2 parents. मेंडल ने जो अपने एक्सपेरिमेंट्स किए थे वो किए थे ऑन अ पी प्लांट। तो उन्होंने क्या कहा कि मैं नाउ आई वांट टू सी हाउ डू दीज जीन्स गेट ट्रांसमिटेड। जैसे यहां पर मैं आपको कह रहा हूं कि यील्ड की जीन हो सकता है यहां से आ जाए, यहां से आ जाए, पेस्ट रेजिस्टेंस की हो सकता है यहां से आ जाए, यहां से आ जाए। तो ये काम कैसे करता है, हाउ डज दिस ट्रांसफर वर्क, दिस इज व्हाट ही वांटेड टू स्टडी। सो फॉर स्टडिंग दिस ही चोज अ पी प्लांट, राइट। व्हेन ही चोज अ पी प्लांट, he said that I will take a plant that is identical in all characteristics. उन्होंने 7 characteristics of pea plant study किए। तो अब इन्होंने क्या किया, उन्होंने 2 pea plants लिए which were identical in 6 characteristics. ये जो 2 हैं, let's call this P2, let's call this P1, they are identical in 6 characteristics. Only difference between these 2 plants is height. This is the only difference. ठीक है, तो अब जो इनका प्रोजेनी होगा, जो इनका चाइल्ड होगा, लेट्स कॉल दिस F1. F1 मींस फीलियल, फीलियल मींस चाइल्ड, सो फीलियल 1 जनरेशन, लेट्स कॉल दिस F1. अब ये जो F1 जनरेशन का पी प्लांट होगा, इसको बाकी जो भी ये 7 कैरेक्टरिस्टिक्स हैं, लेट्स से शेप ऑफ सीड, कलर ऑफ सीड, कलर ऑफ प्लांट, यील्ड ऑफ प्लांट, पेस्ट रेजिस्टेंस, ये सारी चीजें दोनों से मिलेंगी। बट क्योंकि दोनों की सेम है, P1 एंड P2 आर एग्जैक्टली द सेम, तो इसकी भी ये सारी चीजें दोनों से सेम होंगी। ओनली डिफरेंस वी विल सी इज इन टर्म्स ऑफ हाइट ऑफ दिस पी प्लांट, है ना। वेयर डू वी गेट आर डीएनए, वेयर डू वी गेट आर क्रोमोसोम्स, वी गेट 23 पेयर्स, यहां देखते हैं, वी गेट 23 पेयर्स फ्रॉम आवर मदर. This is the universal symbol for females, by the way. We get 23 pair for males. This is universal symbol for males. ठीक है, तो अभी क्या हो रहा है, लेट्स से मेरी जीन्स की बात करते हैं. सो आई हैव गॉटन, मेरा क्रोमोसोम स्ट्रक्चर जो है वो पेयर्स में है. तो मेरे क्रोमोसोम्स कैसे हैं, माय क्रोमोसोम्स आर लाइक दिस, देयर आर 23 पेयर्स. क्रोमोसोम पेयर 1, जो पेयर 1 है उसमें I would have gotten 1 of these from my mother. I would have gotten 1 of these from my father. है ना, ये बात तो आपको समझ आ रही है. 23 from mother, 23 from father, total 23 pairs. तो my mom would have given 1 chromosome of pair 1, जो नंबर 1 है, और मेरे फादर ने क्रोमोसोम नंबर 1 दिया होगा, और अब मेरी बॉडी में इसका पेयर बना हुआ है. अब यहां पर भी कुछ जीन्स हैं, there are some genes here, there are some genes here, है ना. Let's say the gene for color of hair, ठीक है, let's say it's not true, but let's say gene for color of hair, which color hair would get expressed is here. अब यहां से this is the gene for color of hair that I have gotten through my mother. This is the gene for color of hair that I have gotten through my father. अब मेरा कलर ऑफ हेयर क्या होगा, will it be similar to my father, will it be similar to my mother, will it be a mixture of both? ये क्वेश्चन आ रहा होगा आपके दिमाग में, है ना. My mom does not have blue hair, but let's say for now that my mother has, let's say she has blue hair and my father has black hair. ठीक है, अब मेरे हेयर कलर क्या होगा, ब्लू होगा या ब्लैक होगा, राइट? दिस हैज टू बी सीन हियर। अब ये चीज काम कैसे करती है, दिस इज व्हाट मेंडल स्टडीड। जो बोर्ड पे लिखा है ये चीज आपको समझ आ रही है कि क्या मैं बात कर रहा हूं, ये समझ आ रहा है आपको? दिस मस्ट बी क्लियर टू यू, वी हैव डिस्कस दिस मल्टीपल टाइम्स, है ना? नाउ मेंडल हैड टू स्टडी दिस। अब ये स्टडी करने के लिए ह्यूमंस में, आप दो ह्यूमंस को तो रिप्रोड्यूस करा नहीं सकते जस्ट फॉर द पर्पसेस ऑफ डूइंग दिस एक्सपेरिमेंट, है ना? So he decided to do this in pea plants. Whatever mechanism in pea plants will also work in humans because principle of continuity of DNA. तो अब यही चीज इन्होंने, सेम चीज यहां पर भी कलर ऑफ हेयर डिस्कस हो रहा था, अब यहां पर हाइट डिस्कस हो रही है, सेम थिंग। दिस प्लांट, दिस इज पेरेंट 1, दिस इज पेरेंट 2, लेट्स से पेरेंट 1 was tall, so it is giving the genes of tall plant. Parent 2 was dwarf, so it is giving genes of short. अब इनमें से क्या एक्सप्रेस होता है देखते हैं। अब हाइट को प्रोजेनी में करते हैं, F1 he had the first generation F1, यहां पर क्या हुआ, they got a tall plant, ठीक है? तो अब now this was confusing, तो ऐसे इनके he had two such plants, दोनों ही tall आए, F मतलब दो बच्चे कराए इसने टी प्लांट्स के, both were tall, so two tall plants were gotten. फिर मेंडल साहब ने क्या किया, मेंडल साहब ने फिर दोबारा एक्सपेरिमेंट किया एंड ही हैड दीज टू टॉल प्लांट्स ब्रीड विद ईच अदर। तो ये जो F1 में यहां जो ये दो टॉल प्लांट्स बने, दीज बिकेम द पेरेंट्स ऑफ द नेक्स्ट जनरेशन। तो इन्होंने इन दो टॉल प्लांट्स को, लेट मी कॉल इट टॉल प्लांट ऑफ F1 जनरेशन, ही मर्ज देम विद टॉल प्लांट ऑफ F2 जनरेशन। फिर इनको आपस में इन्होंने हाइब्रिडाइज कराया, आपस में रिप्रोड्यूस कराया। अब देखते हैं क्या हुआ। Now 4 plants were produced, 3 were tall, 1 was dwarf. अब ये तो बहुत ही कंफ्यूजिंग हो गया। अब दिस इज वेरी कंफ्यूजिंग। अगर ये सिर्फ टॉल आता तो हम कह देते भाई कि अगर टॉल और ड्वार्फ प्लांट हैं, इफ देयर इज अ टॉल प्लांट एंड देयर इज अ ड्वार्फ प्लांट, every time the child will have tall genes. This I could have said this, but once then this happened, here I have two tall parents, I got 3 tall plants, 1 dwarf plant. How to explain this? So he explained this by his first law called law of dominance. बस यही लॉ समझाऊंगा आपको। अगर आपको ये समझ आ गया, सब कुछ हो गया, तो कल हम ये लॉस डिटेल में देखेंगे। लॉ ऑफ डोमिनेंस, राइट? लेट्स से, चलो वन क्वेश्चन आई विल आस्क यू, लेट्स से देयर इज अ जीन फॉर हाइट, हाउ मेनी कॉपीज ऑफ दैट जीन विल वन पी प्लांट हैव? मान लो कोई एक जीन है जो हाइट कंट्रोल करती है, हाउ मेनी कॉपीज ऑफ दैट जीन, हाउ मेनी अलील्स कहते हैं, दिस इज कॉल्ड अलील्स, बट अभी आप कॉपी समझ लीजिए, हाउ मेनी अलील्स ऑफ दिस जीन वुड अ पी प्लांट हैव? बताओ, सोच के बताना आराम से, वन पी प्लांट में हाइट की कितनी जीन्स होंगी? इफ यू गिव दिस आंसर राइट, you would have understood. 2, very good. Obviously 2, one from the mother, one from the father. तो मेंडल ने क्या एक्सप्लेन किया, उन्होंने एक्सप्लेन किया लॉ ऑफ डोमिनेंस। आई होप यू आर राइटिंग दिस। उन्होंने कहा कि देखो, हाइट की जो जीन्स हैं, द जीन्स ऑफ हाइट कैन बी रिटन एज, दिस इज जीन टी, कैपिटल टी, जीन फॉर टॉल प्लांट। सो दिस इज वन ऑफ द पॉसिबल कॉपीज। अगर हाइट की जीन्स की बात करें तो या तो आपके पास ये जीन हो सकती है कैपिटल टी, जीन फॉर टॉल प्लांट, या आपके पास जीन हो सकती है स्मॉल टी, जीन फॉर ड्वार्फ प्लांट। तो अगर कोई प्लांट ऐसा है जिसके पास मदर ने भी कैपिटल टी दी, फादर ने भी कैपिटल टी दी, दिस वुड बी अ टॉल प्लांट। अगर कोई प्लांट ऐसा है, मदर हैज गिवन स्मॉल टी, फादर हैज गिवन स्मॉल टी, this would be a dwarf plant. Question was, what happens when this happens? Mother has given capital T और father has given capital T and other parent has given small t. फादर से ये जीन मिली, मदर से ये जीन मिली. अगर दोनों से कैपिटल टी मिली तो टॉल, दोनों से स्मॉल टी मिली तो डॉर्फ. बट व्हाट अबाउट दिस? ये प्लांट कैसा होगा? तो ही सेड द कैपिटल टी जीन, ये जो है, दिस इज द डोमिनेंट जीन एंड इट डोमिनेट्स. जब ऐसा कंडीशन होता है, द कैपिटल टी जीन डोमिनेट्स. तो अगर ऐसा भी किसी का जीन स्ट्रक्चर है कि फादर हैज गिवन कैपिटल टी, मदर हैज गिवन स्मॉल टी, इफ दीज आर द टू जीन्स ही हैज, इट वुड स्टिल बी टॉल एंड जस्ट एज टॉल एज दिस मच. ऐसा नहीं है कि इस टॉल और इस टॉल में कोई फर्क होगा. आप देख के नहीं बता सकते दैट दिस इज कैपिटल टी स्मॉल टी, दिस इज कैपिटल टी. दोनों इक्वली टॉल होंगे. अब क्यों हो रहा है ऐसा? We know that there are two strands of DNA, one given from my mother, one from my father. But will both be used? Will both be used when when DNA has to form mRNA? No, one of these will be used. Which will be used? या तो ये जीन यूज होगी या ये जीन यूज होगी. उन्होंने क्या कहा? that there is a dominant gene, कैपिटल टी डोमिनेंट जीन है. अगर ये दोनों जीन्स आ गई तो कैपिटल टी का प्रोटीन बनता है और टॉल बनता है. यहां पर भी कैपिटल टी का प्रोटीन बन रहा है. तो चाहे आपकी जीन्स ऐसी हो, चाहे आपकी जीन्स ऐसी हो, आप टॉल दिखोगे. ये बात अभी तक क्लियर है? आई नो इसका लॉजिक में समझाऊंगा, बट अभी तक ये समझे यहां पे बात क्या हो रही है. देन वी विल पुट दिस ऑन द एक्सपेरिमेंट एंड अंडरस्टैंड. बट ये बात क्लियर है? ये समझ आया यहां पे क्या हुआ? अब इफ दिस वाज हिज थ्योरी, ये इनकी थ्योरी है. लेट अस अप्लाई, दिस इज थ्योरी, दिस इज द थ्योरी ही गिव, द हाइपोथेसिस ही गिव. दिस इज द एक्चुअल रिजल्ट. ये तो हमें फॉर अ फैक्ट पता है ये होता है, ये आप टी प्लांट्स में करके चेक कर सकते हो. सो लेट अस नाउ सी इफ दिस रिजल्ट होल्ड्स इन दिस. उनकी थ्योरी अब इस पे अप्लाई करके देखते हैं. सो व्हाट डिड ही से? दैट पहले देयर आर टू प्लांट्स, वन इज टॉल, वन इज ड्वार्फ. तो इन्होंने कहा पहले ये प्लांट्स ऐसे रहे होंगे, कैपिटल टी इंटू स्मॉल टी. ये टॉल प्लांट होगा, ये ड्वार्फ प्लांट होगा. इनके आप बच्चे कराओगे तो ये कौन सा जीन दे सकता है? सिर्फ कैपिटल टी दे सकता है. दिस कैन ओनली गिव स्मॉल टी. तो इनके जो बच्चे होंगे, दे विल हैव दिस. बोथ किड्स विल लुक लाइक दिस. सो बोथ किड्स इन F1 जनरेशन आर टॉल. सो फार द थ्योरी इज राइट. अभी तक थ्योरी में कोई गलती नहीं है. अब इन्होंने कहा लेट्स ब्रीड दिस टू टुगेदर. सेकंड स्टेप यही था, इनको आपस में पेरेंट्स बना के देखते हैं. इनको पेरेंट्स बना के देखा, इनको पेरेंट्स बना के देखा तो क्या हुआ? नाउ व्हाट आर द पॉसिबल जीन्स दैट दिस कैन गिव? इट विल डू मियोसिस, क्रोमोसोम्स विल सेपरेट, इट विल गिव ओनली वन पेयर, राइट? गमीट्स बनाएगा. गमीट्स बनाएगा तो अब एक डीएनए स्ट्रैंड पे इसके कैपिटल टी है, एक डीएनए स्ट्रैंड पे स्मॉल टी है. गमीट्स बनाएगा तो ऐसे डिवाइड होंगे. एक गमीट में या एक स्पर्म में, लेट्स से, ऑल्दो स्पर्म इज नॉट द गमीट इन केस ऑफ प्लांट्स, एक स्पर्म में कैपिटल टी देगा, एक स्पर्म में स्मॉल टी देगा. इफ दिस इज द फीमेल, दिस आल्सो हैज समथिंग लाइक दिस. एक स्ट्रैंड पे कैपिटल टी, एक स्ट्रैंड पे स्मॉल टी. तो एक एग में कैपिटल टी जाएगा, एक एग में स्मॉल टी जाएगा. यही होगा. अब इसके पॉसिबल कॉम्बिनेशन देख लेते हैं. फ्रॉम द मेल वी आर गेटिंग कैपिटल टी एंड स्मॉल टी. From the female, we are getting capital T and small t. So 4 plants will become, 4 possible combinations are there. What are the 4 possible combinations? Capital T, दोनों के capital T बन गए. These two gametes merged and formed the egg. Second chance is capital T, small t. This gamete merged with this gamete. Now this we can't control. Which sperm wins the egg? We don't. Which sperm wins the race? We don't know. So we are checking all possible combinations. ये और ये अगर गमीट मिल गए तो ऐसा प्लांट बनेगा. दिस विल ऑल्सो बी अ टॉल प्लांट, दिस विल ऑल्सो बी अ टॉल प्लांट. एक क्या पॉसिबिलिटी है? दैट ये और द वन पॉसिबिलिटी इज दैट दीज टू बिकम टुगेदर. ये दोनों गमीट्स मिल जाएं तो कैपिटल टी स्मॉल टी, ये भी टॉल प्लांट है. लास्ट पॉसिबिलिटी इज दैट दीज टू गमीट्स कम टुगेदर. तो ये बनेगा स्मॉल टी स्मॉल टी। तो अब देखो, अब प्लांट्स कैसे दिखेंगे? हाउ विल द प्लांट्स लुक लाइक? 1 tall plant, 2 tall plant, 3 tall plant, 4th dwarf plant. This was his law of dominance. समझ आया? How genes are being controlled. Law of dominance. ठीक है, दिस इज द फर्स्ट मेंडेलियन लॉ। ठीक है, मोर ऑन दिस टुमारो, बट इफ यू हैव अंडरस्टूड दिस, कल मैं आपको स्लाइड्स में भी लिख के दूंगा ये, कल हम करंट अफेयर्स भी काफी सारे देखने वाले हैं। लॉ, ही गेव मेनी लॉज, लॉ ऑफ डोमिनेंस, लॉ ऑफ सेग्रीगेशन, लॉ ऑफ इंडिपेंडेंट असॉर्टमेंट। अब ये जो लॉ ऑफ इंडिपेंडेंट असॉर्टमेंट है, थर्ड लॉ, इसको वायलेट करता है समथिंग कॉल्ड एक्स्ट्रा क्रोमोसोमल डीएनए फाउंड इन ह्यूमंस इन कैंसर सेल्स, विच इज इन द न्यूज। उसमें देयर बी सी समथिंग कॉल्ड द जैकपॉट इफेक्ट, राइट? न्यूज में भी है, मुझे सर ने पढ़ाया भी है। we'll look at it in detail, but I wanted you to understand this law. Good that you've understood, right? Good that you've understood. Now अब चलते हैं आगे। तो obviously now you would understand, because of this uncertainty and till the times genes were not understood, if I am breeding two plants together with beneficial characters, if I am doing something like this, one with good yield, one with pest resistance, if I am breeding them together, there is no certainty that I will get the good gene, right? Which one will go in the gamete, which one is the dominant gene, which, how will that it get transmitted, will it become, will it have both capital T's or capital T and small t, ये नहीं पता मुझे. So this is the uncertainty that was there in traditional hybridization. Just one more piece of information, यहां पर जो हम ये पढ़ रहे थे, कैपिटल टी एंड स्मॉल टी, व्हेन दिस हैपेंस, कैपिटल टी स्मॉल टी, वी गेट अ टॉल प्लांट because capital T is the dominant gene. This was his law, law of dominance. What is small t called? Small t is called the recessive gene. Although कल ये slide में लिखा हुआ मिलेगा आपको, but just for information I'm telling you, small t is called the recessive gene. Just for information. Anyway. अब देखते हैं. So I knew that here, if I'm breeding two plants in traditional hybridization with good characters, there is no guarantee that I would get a beneficial character in the child, because how the genes get separated, which is dominant, etc., I don't know. Right? So there was a certain disadvantage which we have seen. Up. Now we understood all of these things happened. We understood how genetics works, and genetic hybridization or traditional hybridization was replaced with genetic modification, which we are going to see today. But first, I want you to write a mains question. Right? I will give you 3 minutes to think about this. ठीक है, मैं कोई हिंट नहीं दूंगा आपको, आपके पास 3 मिनट से, चैट ऑफ रहेगी, ट्राई करना, जस्ट थिंक, डोंट राइट एनीथिंग, जस्ट थिंक, व्हाट आर द इश्यूज ऑफ एग्रोकेमिकल बेस्ड एग्रीकल्चर? नो हिंट्स, एग्रोकेमिकल का बता देता हूं मीनिंग। एग्रोकेमिकल मींस पेस्टिसाइड्स, फर्टिलाइजर्स, एक्सेट्रा। इसके इश्यूज क्या हैं? 3 मिनट है, सोचो, जस्ट थिंक, व्हाट पॉइंट्स विल यू राइट इफ यू सी दिस इन द मीन्स? थिंक, सोचना है, as many points as you can think of. आंसर, इंट्रोडक्शन, कंक्लूजन मत सोचना, बॉडी के पॉइंट्स सोचना, हाउ, व्हाटएवर मेनी पॉइंट्स यू कैन थिंक ऑफ. 1 minute left. सोचो सोचो. As many points as you can generate. और सिर्फ it only think about the body. Don't think about intro or conclusion. Intro conclusion बात की बात होती है. लास्ट 20 सेकेंड्स. जनरली मीन्स में आपको इस काम के लिए, जो काम मैं आपसे अभी ये करवा रहा हूं, इस काम के लिए आपको मीन्स में 1 मिनट मिलता है. आपको मैंने 3 मिनट दिए. सो दिस इज 3 times the time you will get in your actual means paper. Maximum you will get 1 minute to think because you will have to write also. 6 minutes will go in writing. ठीक है, बताओ जी क्या सोचा आपने. ठीक है, गुड. देखो, I've read most of the comments that have come so far. Almost all of them are very good. सारे comments, सारे points valid है. Now what should this tell you? और आपकी chat public है तो आप भी देख सकते हो. Thinking this answer is not a difficult thing. अगर आपके points अच्छे हैं, it's not a good thing. Learn to recognize this fact that अगर आपके competitors के भी points अच्छे हैं, everybody sitting in this class is your competitor, right? Everybody is trying for the same thing that you are trying. वो अगर आपसे ज़्यादा ध्यान से सुनेंगे, if they are, understand this more clearly, they have greater chances of being selected, right? If all your competitors know the answer to this question, and they all know the points of this question, then it's problematic, no? How will you differentiate? जो आप लिख रहे हो वो बाकी भी लिख रहे हैं. Why will you get more marks? Way you can differentiate here is this approach. अब कैसे अप्रोच लगाते हैं? This is what I do in Write Smart. आपको बस करके दिखा रहा हूं. What are the stakeholders involved in this process? I'm sure इस क्लास में बहुत लोग वो भी होंगे जिन्होंने मेरे साथ Write Smart किया है. अगर तुमने ये stakeholders नहीं सोचा तो I am disappointed in you. Because तुम्हें तो मैं कम से कम ऐसे 50 क्वेश्चंस तो करा चुका हूँ स्टेकहोल्डर्स लिखवा के, है ना? अगर आपने नहीं किया है मेरे साथ, then it's okay. But if you have done this with me and not thought of this, then need to work after prelims. Okay. Anyway, stakeholders कौन कौन है? Farmer is a stakeholder. Government is a stakeholder. Consumer is a stakeholder. Environment is a stakeholder. Farmer को क्या प्रॉब्लम है? कॉस्ट ज्यादा है भाई, फर्टिलाइजर्स की, पेस्टिसाइड्स की. फार्मर की हेल्थ इफेक्ट होती है, right? When he's spraying these pesticides, when he's spraying these fertilizers, there is a health problem. Consumer को क्या प्रॉब्लम है? Since the farmer's cost is high, consumer's cost is also high. These things are not good for the body. They can get bioaccumulated, they can get biomagnified, ultimately have a bad impact on the health of consumers. Government को क्या प्रॉब्लम है? When farmers will use expensive fertilizers like diammonium phosphate, muriate of potash, urea, government will have to import these. When government will have to import these, one, our current account deficit will increase. Two, our subsidy bill will increase. So it will leave less space or less fiscal space for the government to spend on capital expenditure, because there will be more spending on subsidies and revenue expenditure. When the cost of inputs for the farmer is increasing, government will also have to promise a greater level of minimum support price, again increasing revenue expenditure costs, leaving less fiscal space for capex. And lastly, the environment also gets impacted with them, right? Yeah, I'm sure आपने थोड़ा बहुत पढ़ा होगा, although I will teach you all of this in detail. There will be eutrophication, there will be pollution of groundwater also, there will be an impact on soil fertility also. अब देखो यहाँ कितने पॉइंट्स लिखे हैं. 1 2 3 4 5 6 7 8 9 10 11 points are written, all thought of logically, answer appears logical, and it has taken me less than 3 minutes to write it on the board. सोचने में तो मुझे ये शायद 1 मिनट लगता है, 1 मिनट भी नहीं लगता, राइट? दिस इज हाउ यू डिफरेंशिएट. ठीक है, अब आप और सोचते जाओ. ये करंट अकाउंट डेफिसिट में और ज्यादा प्रॉब्लम है, बिकॉज व्हेन वी यूज दीज एग्रोकेमिकल्स वी विल बी subject to sanitary and phytosanitary measures of WTO. So we will not be able to export to European countries. Now there's no limit because you are thinking in a a particular order. समझ आया? This is the difference between how a serious aspirant thinks and how anybody thinks. बाकी चीज तो कोई भी लिख सकता है ना? ये तो अगर मान लो आप किसी नॉर्मल वेल रेड सिटीजन ऑफ इंडिया से पूछो, right, any normal well read citizen of India, for example, I'll relate with my father, right? He reads the newspaper every day. He's not preparing for UPSC and never wants to prepare, right? अगर उनसे मैं पूछता जो पॉइंट्स आपने बताए वो बता देते. How will you differentiate? So always when you read something, try and achieve this. आगे चलते हैं. अब ये प्रॉब्लम्स हैं. These are the problems of agrochemical based agriculture. These problems का 5 problems, 1 solution is GM crops. ठीक है. What can we do? I can take genes from various organisms. I can be sure that I will be able to cut those genes from there, carry those genes and paste them, therefore leading to better characteristics. So I can use genetic modification to make crops tolerant to abiotic stresses. Again, do word breakdown. Abiotic. Biotic is living. Antibiotic, against living things. Which is antibiotic? What is the living thing it is against? Bacteria. Biotic means living. Abiotic stresses, non-living stress. Cold, drought, salt, heat. What would be biotic stress? Pests, bacteria, herbs, weeds. So you can reduce your reliance on chemical pesticide. You can reduce post-harvest losses because everything must be controlled through genes only. You can increase the efficiency of mineral usage by plants, right? How efficiently they are able to use the minerals. They will get, let's say, 10 micrograms of nitrogen. How efficiently can they use this? This also depends on their genes. If they are able to use minerals efficiently, soil fertility will not deplete. अगर वो 1 gram of nitrogen में अपना पूरा काम चला लेता है, soil has 10 grams, तो 1 gram में काम चलेगा. If it is not efficient, it will need 5 grams. So it will have an impact on fertility of soil. You can increase their nutritional value. For example, you can put the gene of vitamin A inside rice. Rice you will eat, you will also get vitamin A with them. Normally rice don't contain vitamin A. These are known as golden rice. ये भी कर सकते हो, right? And you can also do this. Tailor-made plants for certain products. GM alga for biofuels. This is what we will read when we read environment. There are 4 generations of algae and biofuels that we'll read. ये हम तब पढ़ेंगे, but we can do this also. These are the advantages of GM crops. ये आपके मेंस में बहुत काम आएगा आपको, because GM crops पे क्वेश्चन आ चुका है. अब एक एडवांटेज थी पेस्ट रेजिस्टेंस. पेस्ट रेजिस्टेंस का एग्जांपल मैंने आपको बताया था कि मैं बैक्टीरिया से एक जीन उठाऊं और उसे कॉटन प्लांट में डाल दूं. रिमेंबर वी हैव स्टडीड दिस एग्जांपल. दिस इज द एग्जैक्ट स्टोरी ऑफ बीटी कॉटन. नाउ लेट्स रीड इन डिटेल. सम स्ट्रेन्स ऑफ बैसिलस थ्यूरिंजेंसिस. बैक्टीरिया का नाम है, बैक्टीरिया का नाम है बैसिलस थ्यूरिंजेंसिस. सम स्ट्रेन्स ऑफ बैसिलस थ्यूरिंजेंसिस प्रोड्यूस प्रोटीन्स दैट किल सर्टेन इंसेक्ट्स. अब इसमें क्या होगा? ये बैसिलस थ्यूरिंजेंसिस मींस बीटी. दिस बीटी would have a gene. This gene would create a protein which would be, let's say, an enzyme that would kill bollworms when they eat it. तो bollworm को बीटी हो रही है. जिसको समझ आ गया ठीक है, जिसको नहीं आया कोई बात नहीं. ठीक है. Now, so this particular bacteria must have a gene that's coding for a protein that then makes an enzyme, or that protein is the enzyme that kills the bollworm. अब मेरा आपसे क्वेश्चन क्या है? इसका मतलब क्या हुआ? This tells me that this particular enzyme is a very toxic enzyme. The moment the bollworm eats it, the bollworm dies. So enzyme is fairly toxic, right? It can be almost poisonous. Why does the bacteria not die? बैक्टीरिया के अंदर ही एंजाइम एक्सप्रेस हो रहा है, बैक्टीरिया के अंदर ही एंजाइम है. बैक्टीरिया itself does not die because it is not made in the form of toxin. यहां पर टॉक्सिन, enzyme that is toxin is called toxin. It is not made in the form of toxin, it is made in the form of, अब आपको दोबारा वर्ड ब्रेक डाउन करना है, प्रोटॉक्सिन. अब प्रोटॉक्सिन का क्या मतलब बायोलॉजी में? प्रोकैरियोट, ओल्डर देन न्यूक्लियस, एंशिएंट न्यूक्लियस, बिफोर टॉक्सिन. तो इन बैक्टीरिया इट एग्जिस्ट एज प्रोटॉक्सिन. ये बैक्टीरियल सेल है, यहां पर ये एंजाइम पड़ा है, ये यहां पर प्रोटॉक्सिन है. दिस इज प्रोटॉक्सिन. व्हेन द बॉलवर्म विल कम एंड ईट इट, जब बॉलवर्म ने आके ये खाया, इनसाइड द बॉलवर्म इट गेट्स कन्वर्टेड फ्रॉम प्रोटॉक्सिन टू टॉक्सिन and the bollworm dies. तो अब क्या होगा? There must be some gene inside this Bacillus thuringiensis that is coding for this enzyme, and this enzyme is a protoxin, goes inside bollworm dies. I will take this gene from Bacillus thuringiensis and give it to cotton. So I'll just read now, remains as a protoxin in bacteria, becomes ingested means eaten by bollworm, it becomes toxin, right? Bt toxin gene has been cloned from bacteria and expressed in plants. जीन उठाई बैक्टीरिया से, प्लांट्स में एक्सप्रेस कर दी to provide resistance to insects without the need for insecticides. Now I don't need chemicals. Specific Bt toxin genes isolated from bacteria and incorporated into plants. Now choice of the gene depends on crop. कौन से क्रॉप, कौन सा पेस्ट वगैरह. दिस ये मैं आपको पढ़ा चुका हूं. दिस हैज क्रिएटेड अ बायोपेस्टिसाइड. एग्जांपल्स बीटी कॉटन, बीटी कॉर्न, बीटी राइस, बीटी टमेटो, ऐसे बहुत सारे हैं. इंडिया में ओनली बीटी कॉटन इज अलाउड. नाउ व्हाट इज द नेम ऑफ दिस जीन? बैक्टीरिया इज कोडिंग फॉर अ पर्टिकुलर जीन. बैक्टीरिया इज कोडिंग फॉर अ पर्टिकुलर जीन. I am taking this gene and I am putting this inside a plant. Now plant will also make this particular protoxin. The moment bollworm comes and eats it, bollworm will die. The name of this gene is cry gene. Cry, C-R-Y, cry. ठीक है. अब names are cry1AC, cry2AB. These are for cotton bollworms. cry1AB is for corn borer. So for corn, for cotton वहां यूज कर सकते हैं. अब यहां पे लिखा है Bacillus thuringiensis. There is a gene I isolate, I incorporate into cotton. Now cotton plant also produces protoxin. Now protected against bollworms. Right? So this is BT cotton and bollworm. Concept of BT cotton. Is this clear? कि BT cotton में हो क्या रहा है? पढ़ चुके हैं वैसे पहले, but now I think would be becoming even more clear to you. Just taking a gene from there, putting it here. है ना? Fair enough. ठीक है. आगे एप्लीकेशंस देखते हैं. Second method is RNA interference. Now this will take a little bit of more understanding. थोड़ा टाइम लगेगा इसको अंडरस्टैंड करने में, बट ठीक है. बेसिकली आई टेल यू द प्रॉब्लम स्टेटमेंट. देखो साइंस में सारी रिसर्च ऐसी होती है. देयर इज अ प्रॉब्लम स्टेटमेंट. यू नो कि अच्छा यार ये प्रॉब्लम है. आई टेल यू व्हाट द प्रॉब्लम स्टेटमेंट वाज, देन आई टेल यू व्हाट वी डिड टू सॉल्व दैट प्रॉब्लम, देन यू विल अंडरस्टैंड इट इवन बेटर. ठीक है. तो मेथड छोड़ देते हैं. लेट्स फर्स्ट अंडरस्टैंड हो क्या रहा था. हमने एक नेमाटोड पढ़ा था आज से पहले, सी एलिगेंस, इन केस ऑफ द नोबेल प्राइज. तो हमने क्या देखा, देयर वाज अ नेमाटोड कॉल्ड मिलियोडाइन इनकॉग्निशिया. बिल्कुल नाम याद रखने की जरूरत नहीं है, बट स्टिल जस्ट फॉर रेफरेंस, एम इनकॉग्निशिया नाम का एक नेमाटोड था. दैट वाज इन्फेक्टिंग रूट्स ऑफ टबैको प्लांट्स एंड कॉजिंग अ ग्रेट रिडक्शन इन यील्ड. दिस इज द प्रॉब्लम स्टेटमेंट। ठीक है, ये देख रहे हो आप, ये दिस इज डायरेक्टली फ्रॉम 12th क्लास एनसीईआरटी बुक। ये देख रहे हो ऐसी दिखने लगती थी रूट्स। दिस सॉर्ट ऑफ इन्फेक्शन इज देयर इन द रूट। यू कैन सी दिस bulbous appearance of roots. After this, roots were not able to absorb not able to absorb water or nutrients from the ground. Yield reduced drastically. This was the problem. The problem statement क्या है? M. incognitia नाम का एक nematode है. This infects the tobacco plant and causes a great reduction in yield. अब इस प्रॉब्लम को सॉल्व करना है। ऑब्वियसली देयर इज वन वे कि आप पेस्टिसाइड्स दे दो जिससे ये मर जाए, बट अगेन द केमिकल एग्रीकल्चर प्रॉब्लम्स कम इन। सो वी हैव टू नाउ अंडरस्टैंड हाउ टू सॉल्व दिस प्रॉब्लम, राइट? हाउ वाज बायोटेक्नोलॉजी यूज्ड टू सॉल्व दिस प्रॉब्लम? लेट्स अंडरस्टैंड। फर्स्ट लेट्स अंडरस्टैंड व्हाट इज हैपनिंग। दिस इज, लेट्स से, द रूट ऑफ टबैको। उसका एक सेल है ये। One cell of this tobacco root is one cell. What is happening now? There is a nematode by the name of M. incognitia. It is coming and infecting this. How would it be infecting? It would be entering, then expressing from its DNA, expressing some mRNA, from this making some enzymes. This enzymes would be infecting and taking over or causing some sort of issue with this cell. यही हो रहा होगा ना? What else could happen? How else can anything else get expressed? Now you have to stop this MI, melodyne incognitia, from infecting this root of tobacco. यही कहानी है. You can stop it at anywhere in this process. You can probably stop it from entering the cell. You can stop it from making this mRNA. You stop it from making mRNA to protein. You stop the enzyme from working. आप कोई भी स्टेप पे अगर स्टॉप कर दोगे तो ये प्रॉब्लम सॉल्व हो जाएगी. This is how medicines work, by the way. Any medicine that works, you first identify कि अच्छा यार ये हो रहा है. This thing happens in the body also. Some bacteria is infecting us, etc. Then we see what is the process of that bacteria. Then we find the most easy step where we can stop this entire mechanism from working. Wherever we can find, whichever is the easiest step where we can find a mechanism to stop this process, we make a medicine for that process. This is exactly what we do in any medicine that's formed. So again, here we're looking for a medicine to be formed, right? Now, it was identified that this process can be stopped in the tobacco root cell. This process of mRNA to enzyme, we can stop this process. It's easy to stop this process. How do we stop it? Let's look at this process in a bit more detail. What would be happening here? There would be a double helix sort of DNA. From here, a single strand of mRNA would be made, and then some enzyme would be made. यही हो रहा होगा. We said that we will not let this mRNA make the enzyme. तो हमने क्या किया? Now this is the root cell of tobacco. It has its own nucleus. It has its own chromatin. ये tobacco का DNA है ना? हमने क्या किया? Somehow, how we'll talk about. Somehow we introduced these genes. अब यहां पर कोई जीन होगी जो mRNA बनाती होगी. Let's say this is gene number 1, G1. ठीक है? We introduce this gene number 1 into this DNA. We identified कि अच्छा यार इस MI की कौन सी जीन थी which makes the most harmful enzyme, and we put that gene inside this. Then we modified the cell in such a way that whenever MI infects from this gene, from this G1 that the tobacco itself has, ये tobacco का genetic structure है, ये MI का genetic structure है. Tobacco would also make an mRNA from this gene. Obviously, little bit more complicated than I'm explaining to you. I'm trying to make it as simple as possible. Tobacco would also make an mRNA. This mRNA would bind to this mRNA. So what would happen? ये mRNA का एक strand बनेगा ऐसा. ये जाके यहां ऐसे bind कर जाएगा. Once it goes and binds, once it goes and binds, can then the enzyme be created? Can double-stranded RNA like this, when it is bound, can this be translated? No. We have read this. Same kahani was happening with microRNA. Post-transcriptional gene regulation was happening. यहाँ भी वही काम हो रहा है. We are effectively, but this is happening in different organisms, so we can't call this gene regulation. ये एक और organism है, एक दूसरा organism है. This is just defense by the cell. The root cell is defending itself from this process by making a mRNA that is complementary to this mRNA, binds with this mRNA, and stops this process of enzyme formation. Basically, we have interfered with the mRNA, not let this mRNA form enzyme and then lead to this issue. So this is called RNA interference. Basically, I have made the tobacco cell. I have made the tobacco cell immune. मैंने tobacco cell में एक protection mechanism डाल दिया है against this MI. I have put in the tobacco cell a protection mechanism against MI. I can call this a type of, आज हम पढ़ेंगे ये vaccination, type of plant vaccine. Yes? समझ आया क्या हो रहा है RNA interference में? I am interfering with the RNA. अब अगर आपको बेसिक प्रोसेस क्लियर है तो दिस शुड बी वेरी क्लियर टू यू. अगर आपने बेसिक्स रिवाइज किए हैं तो इसमें कोई दिक्कत ही नहीं आनी चाहिए. तभी आपको कल टाइम दिया था कि आप बेसिक्स रिवाइज कर लो. सो लेट्स सी व्हाट ऑल हैज हैपेंड. I was seeing or there was a problem that we were seeing, that there is a nematode by the name MI that infects tobacco cells, converts its DNA into mRNA, makes an enzyme that creates an issue in the root cells and greatly reduces the yield. This was my problem. Now I have to stop this process somewhere. This process of DNA to RNA to enzymes to issue, this process has to be stopped, otherwise it will keep on creating this issue. तो मैंने क्या किया? I decided that I will stop this process at this stage. I will stop this mRNA to enzyme happening. How did I stop this? I introduced some genes which are nematode related genes. Obviously this is a nematode gene. एक nematode DNA होगा ये, जिससे ये mRNA बनता होगा. So I introduced some nematode based genes in the tobacco. I introduced nematode genes in the tobacco here. Tobacco plant है ये. Tobacco plant is expressing an mRNA now. Whenever this MI infects, it makes its own mRNA which is single stranded. From here, it has this gene, it makes this mRNA which is single stranded. Tobacco plant sees MI has come. Tobacco plant also makes mRNA which is from nematode genes. These mRNA two join together like this. After this joining, because of the double-stranded nature of this mRNA, it does not form enzyme. It does not form enzyme, there is no issue. Basically, we have interfered with the RNA of the nematode, therefore called RNA interference. We have given the tobacco plant a sort of defense mechanism against this nematode. We have created immunity in the tobacco plant, therefore called or referred to something close to a plant vaccine. If we do this for plants, known as plant vaccine. Now, what was the difficult step here? What was the difficult step? Difficult step here was finding these genes, finding these nematode genes and giving it to tobacco plant, right? Because can I insert DNA directly in a cell? Can I just inject DNA? कर सकता हूं क्या? No. We have read that if I just inject DNA, अगर मैं सिर्फ, if I just inject DNA, it will not replicate. है ना, क्यों नहीं रेप्लिकेट करता उसमें नहीं जाते हैं, बट इफ आई जस्ट इंजेक्ट डीएनए डायरेक्टली इट विल नॉट रेप्लिकेट. I read this yesterday that I need a carrier. Carrier also known as vector. I need a vector for this gene to go. All clear now? अब पढ़ लेते हैं, समझ आ जाएगा. All clear? आसान होती है यार साइंस, बस बेसिक क्लियर होने चाहिए. This is not something I have to teach you. You could have understood this yourself also. But you should know the basics. Basics, it was important that I teach you. अब पढ़ते हैं. These lines are directly from the NCERT, okay? ताकि we know that we are reading from authentic, directly copied from the NCERT. Most of these lines I don't write. Where I write, I'll tell you. Main idea here is to silence the mRNA of the pathogen. अब हाँ, one word has come here, very important word, pathogen. Gen means generating. What was MI generating? MI was generating a disease. Patho means disease. In biology, word patho means disease. So study of disease, pathology. This is a branch of medicine who study. In pathologist का क्या काम होता है? आपने देखा होगा path lab. You must have heard path labs. जो ये labs होती हैं where you give your samples for disease testing. They are called pathology labs or path labs because their job is to study samples on a slide and try and identify if any particular disease causing organism is there, if there is any disease pattern is there in the cell. Cell disease होता है तो अलग तरीके से दिखता है. This is their job. So pathology is disease. Pathogen, disease generating. Pathogen, disease generating. Who is disease generating here? MI is disease generating. mRNA, the idea here is to silence the mRNA of the pathogen so that it cannot express its proteins. बात सही है. Method involves silencing of a specific mRNA. Specific mRNA. कौन सा mRNA? MI का mRNA हम silence कर रहे हैं. We want to silence that mRNA. We don't want to let it make proteins. Due to complementary dsRNA molecule. dsRNA, double-stranded RNA molecule. Because of we want the formation of this double-stranded RNA molecule. इस टाइप का हम बनाना चाहते हैं through silencing. Right? That prevents translation of mRNA. mRNA will not get translated. mRNA will not get translated. Protein will not form. A nematode called MI infects the roots of tobacco plant, causes a great reduction in yield. ठीक है? Using agrobacterium vectors. अब देखो, I needed a carrier. I told you I will need a carrier for this process. I will need this carrier. Who is the carrier in this case? Using agrobacterium vectors. Agrobacterium is the name of a bacteria. हमने देखा था plasmid को carrier बना सकते हैं. Vector means carrier. So who is the carrier? Agrobacterium is the carrier. Using agrobacterium vectors, nematode specific genes. मुझे nematode की genes डालनी थी. Nematode की genes, MI की genes plant में डालनी थी. Nematode specific genes were introduced into the host plant. Host plant is tobacco. This allowed the host or tobacco to produce mRNA complementary to the mRNA of pathogen. बिल्कुल allowed tobacco to produce complementary mRNA to pathogen. This orange mRNA complementary mRNA बना पाया. Hence, two RNAs being complementary to each other formed a double-stranded RNA that initiated RNA interference and thus silenced the specific mRNA of the nematode because of the formation of dsRNA due to binding of two strands. One strand, two strand, they bound together, no translation. Because of the formation of dsRNA due to binding of two strands, process of translation was stopped. Good. Consequence was that the parasite could not survive in a transgenic host. Transgenesis मैं आपको डिटेल में पढ़ाऊंगा. For now, transgenesis means genetically modified. Transgenic host expressing specific interfering RNA. अब NCERT में यही लाइन लिखी है. One line we have left. Read the first line. I'll read it with you. राइट? एनसीईआरटी में ये लाइंस लिखी हैं. दैट इज व्हाई if I would have read like this, it probably would not have become very clear. And that's why you need basic understanding. But अब जब basics clear हैं तो आप ये आराम से पढ़ सकते हो. Last line क्या है? Again from the NCERT, RNA interference takes place in all eukaryotic organisms as a method of cellular defense. Not this type of RNA interference, but some sort of RNA interference. The same mechanism is happening in all eukaryotes, not in prokaryotes, as a method of defense of cell. This was, like I told you, the defense of the cell. NCERT में एक दो चीजें और लिखी हैं. This is the process के ये डबल स्ट्रैंडेड आरएनए बन गया. यहां पर जैसे हमारा डबल स्ट्रैंडेड आरएनए बना था, इसे डबल स्ट्रैंडेड आरएनए बन गया. There was binding of double stranded RNA. This was, let's say, mi का mRNA. This was made by tobacco plant. They bound together. Double stranded mRNA is there. Then there are two other proteins involved. You don't need to remember their mechanism of working or formation. You just need to remember their names. A dicer and a risk protein are also involved in this process. What do they do? After this double strand has been formed, अब translation तो रुक गई है, but इसको ऐसे क्यों रहने देना है? What do dicer and risk do? They come and cut this ऐसे. So यहाँ पे दिखाया देखो. This is again from the NCERT. Double stranded mRNA formed. Dicer and risk protein will come and cut this. ऐसे छोटे छोटे पार्ट्स में कट कर देगा, so that there is no chance of this getting expressed. गलती से कहीं बाइंडिंग टूट गई, बाद में दिक्कत हो गई, बाइंडिंग हुई, रिकॉग्नाइज किया, काट दिया. Now mi cannot express itself. So in this process again, if I have to explain this, what will happen? Dicer and risk would come. Dicer and risk would then recognize this and cut it into small pieces. Now we have effectively stopped mi from infecting the tobacco cell, and this is that diagram. This is mi plant. This is mi infected tobacco plant. You see these bulbous formations on the roots. And this is after RNA interference. You do RNA interference, छोटे बहुत कहीं कहीं दिखते हैं, but no major infection. Infection gone. Roots are normal. Yield is normal. These two diagrams are also from the NCERT. RNA interference clear? We just read RNA interference. So RNA interference we have read, BT cotton we have read. ठीक है. अब नेक्स्ट टाइप ऑफ बायोटेक्नोलॉजी देखते हैं, माइक्रोराइजल बायोटेक्नोलॉजी. बट आरएनए इंटरफेरेंस समझ आया? प्लीज रीड द स्लाइड्स वंस. एक बार स्लाइड्स रीड करोगे तो ही समझ आएगा. ठीक है, चलिए आगे चलते हैं. Let us read GM crops and use of mycorrhiza. Although we will read about mycorrhiza in detail when we read environment, but still, let's discuss this in detail once. Mycorrhiza क्या है? Mycorrhiza is a symbiotic relationship. Symbiotic means mutually beneficial. Both organisms are benefiting between a fungus and a root system. Now, what is the main difference between fungus and plants? Fungus cannot generate its own food. Fungus, for example, would eat food from dead and decomposing organisms. You keep bread out in the open, after some time fungus would grow on it and eat that bread. But it cannot generate its own food. So it needs another outer source of food. Plants, on the other hand, can generate their own food, right? So plants can do photosynthesis and they can generate their own food. So this is the main difference between fungus and plants. What is happening here is a fungus called mycorrhiza is associating with a plant. तो क्या हो रहा है कि एक साइड एक फंगस है. This cannot make its own food. It needs a source of food. It is associating with a plant. Plant can obviously make its own food. Now this relationship between these two organizations or two organisms is symbiotic, therefore mutually beneficial. How is the mutual benefit occurring? Plant gives food to the fungus. Very clear. So fungus gets the food it needs. Fungus helps the plant in various ways. It helps the plants in various ways. वो क्या help है वो हम देखते हैं अभी. So a network of fungus filaments. Fungus filaments means fungus जो होता है. Fungus general structure is like this. It is something like this. This sort of growth. These are called fungal hyphae. Please do not go on my drawing. Please Google fungal hyphae and see what they actually look like. Very bad at drawing, but basically this is what it looks like, right? So a network of fungus filaments surrounds the root of the plant. अब ये जो hyphae हैं, ये hyphae इस टाइप के, let's say this is the root and fungus has now surrounded this root and become like this. Now ये जो hyphae हैं, this fungus filaments, they have a larger surface area. So they collect water and mineral from a significantly more amount of soil than the root. So if the root was by itself, अगर root itself होती, right? So if it was just the root, it would look like this. If it is root plus fungus, it's looking something like this. Now the root can collect water from significantly higher amount of plant, so significantly higher amount of soil. This is the help that the plant is receiving, right? So water and minerals from significantly more amount of soil. Then there are certain other benefits. Mycorrhizal plants that have this relationship, have tolerance to adverse soil. Adverse soil meaning soil that does not have good minerals, not enough water, pH, high temperature, drought and toxic heavy metals. इनसे tolerance बढ़ाता है. It also helps plant resist certain diseases. Roots receive, what is the help that the plant gets? Root receives water, more minerals from the fungus. And the mycorrhiza or the fungus receives sugars and nitrogen containing compounds, basically food. So what is happening again? Plant is giving food to fungus. Fungus is giving help to plant through better development of its roots, better tolerance of diseases, tolerance of pests, etc., etc. This is the symbiotic relationship. Now if you use genetic modification to induce this relationship, आप कर सकते हो ना. I'm not explaining the process, but I'm sure it's clear. I can induce this relationship also. This happens normally in nature. Doesn't happen in every plant. Some plants will have the enzymes to bind with fungus. You take that gene and you put it in a different plant, and then you use this technology. You can induce this directly. Again, this is using biology for human benefit, known as mycorrhizal biotechnology. Using mycorrhiza in various plants for the benefits that it gives. ठीक है, should be fairly clear, nothing much to understand. अब ये है difficult thing to understand. ठीक है, GM mustard और धारा mustard hybrid 11. ठीक है, अब देखो, there is an inherent contradiction in the name. Name में ही एक contradiction है. Can you find that contradiction? छोड़ दो यहाँ क्या लिखा है. And notebooks out for this one please. I am saying there is a GM mustard and its name is Dhara Mustard Hybrid 11. There is an inherent contradiction in this name. एक प्रॉब्लम है इस नाम में. क्या दिक्कत है बताओ. Good. If it is a GM plant, why is it hybrid? If it is hybrid plant, it cannot be GM. कुछ तो दिक्कत है. There is some problem. है ना. ये प्रॉब्लम हम देखते हैं ब्रेक के बाद. 4 minutes का ब्रेक. 4 minutes ज्यादा है. 3 minutes का ब्रेक लेते हैं. And after 3 minutes, we will see why is DMH11 called what it's called. See you in 3 minutes. ऑल राइट, अब देखो हाइब्रिड भी है और जीएम भी है. लेट्स सी व्हाट हैपेंस इन अ धारा मस्टर्ड हाइब्रिड 11. ठीक है, दिस इज जस्ट फॉर एक्सप्लेनेशन. जो याद करना है वो स्लाइड्स में लिखा है. दिस इज फॉर एक्सप्लेनेशन. वी विल ब्रेक डाउन एवरीथिंग. व्हाई इज इट कॉल्ड मस्टर्ड, तो क्लियरली व्हाई इज इट हाइब्रिड, व्हाई इज इन 11 इन द नेम एक्सेट्रा. देखो हमने क्या देखा, इन एनी प्लांट if I have to breed that plant, I have to take male, I have to take female. They will both form their gametes. In humans, male gamete is called sperm. In plants, male gamete is called pollen. आपने सुना होगा pollen grains. It is nothing but the sperm of the plant. In humans, female gamete is called egg. Here also, female gamete is called egg or ovary. It's coming from the ovary, it's called the egg. So let's call ovary. From ovary, the female gamete or the egg is coming. अब मैंने देखा that there was two categories of mustard, right? One was called Varuna and one was called Hira. So there was one called Varuna. Varuna is Indian variety, and there was one called Hira. Hira is European variety. There are some beneficial characters in Varuna, some beneficial characters in Hira, and if possibly I could hybridize them. अगर मैं दोनों को मिला दूं, I could get beneficial characters. Somehow it was possible. Obviously, hybridization is not 100% sure, but sometimes you will get beneficial characters also. And here, more often than not, you would get beneficial characters if you could hybridize Varuna and Hira. तो अब बस आराम से hybridize कर लो. What's the problem? Take pollen from male or Varuna, take ovary from Hira and hybridize. Called Dara mustard hybrid. Why do I need GM technology here? The problem in mustard is, and this is what something happens only exclusively in plants, not in humans, it can happen in other organisms. In mustard, both the male and the female sex organs are present on the same plant. So it undergoes a process of self-fertilization. It fertilizes itself. Please don't relate this to humans or you will only laugh and not understand. In plants, it's possible. Okay, there is self-fertilization happening. Male and female sex organs are present on the same plant. अब मैं hybridize करूं कैसे? There is Hira. I want to take, let's say, pollen from Hira and give it to Varuna. But Varuna will get pollen from itself only. जिससे पहले कि मैं pollen लेके आऊं, right? Knowing this condition, this is the plant. It has ovary also and it has pollen also. Before I get the pollen from the other plant and try and hybridize, it will take its own pollen. First, this sperm will always win the race and I will not be able to hybridize. So now I need to stop this pollen from coming. अब एक तरीका तो है कि मैं हाथ से एक pollen निकालूं, वो तो हो नहीं सकता. उसमें भी हो सकता है एक ट्रांसफर हो जाए, right? So I will first need to make sure that this pollen cannot get to the ovary. अभी तक बात समझ आ रही है? I have to make sure that this pollen from here cannot get to the ovary of the same plant so that I can get a different pollen. अभी तक is it clear what's happening? अगर ये clear है तो DMH 11 हो गया. समझ आ रहा है क्या बोल रहा हूं मैं? Good. So what will I do? I will insert a gene. Now I will genetically modify this particular plant. I will genetically modify. What will I do? In this plant, I will introduce a gene called Barnase. Barnase gene leads to male infertility in this plant. Male infertility. What is male infertility? Now the pollen is infertile. The male does not produce fertile sperms. male does not produce fertile pollen. Now this male, this plant cannot give its own pollen because it has become infertile because of Barnase. Infertility happens in humans also, must have heard. Impotency, infertility can happen here also. So I have made the male infertile. I make the male infertile and I get this pollen from the other male here. In this plant, I make the male part. इस plant का ये वाला part मैंने infertile कर दिया and I got a pollen from the other. Now hybridization has happened. Now I have created a hybrid plant. Now I have created Dhara mustard. Dhara mustard has its own egg. Let's say egg coming from Varuna variety. And I have been able to get pollen from Hira variety. Because खुद का pollen infertile हो गया, तो pollen has coming now from Hira variety. I have been able to do this. अब मुझे क्या चाहिए? Now what do I want? Now I want this plant to self-fertilize. Now this plant, this Dhara plant now. अब ये जो धारा बना इसमें क्या है? This Dhara has this sort of arrangement. That ovary, let's say this is the ovary. Ovary is from Varuna. And its own pollen is inactivated, तो ये तो हट गया, right? The pollen is coming from Hira variety. This is what the Dhara plant looks like. Pollen of Hira. Once the kid is produced, the progeny is produced. Pollen is of Hira. Varuna is the ovary. Now this is what I wanted. अब मैं चाहता हूं कि ये सेल्फ फर्टिलाइज करता रहे, हमेशा हीरा का ही pollen आए और वरुणा का ही वो हो. Now I wanted to self-fertilize again, but there is a problem. I have put the gene of male infertility here. मैं पहले ही इसका male infertile इसको कर चुका हूं. अब समझ आ रहा है क्या बोल रहा हूं मैं? Even is this clear? I have gotten the first progeny. पहला hybridization मैंने करा लिया. Now I wanted to keep self-fertilizing. I wanted to keep self-fertilizing now. But there is a problem because already I have put Barnase gene. I have put Barnase gene already, therefore it will not self-fertilize. Still it has a problem of male infertility because of Barnase. So अब problem statement क्या है? ये Barnase एक problem create कर रहा है. Now I will put another gene inside it. I will make another gene enter this called bar star. This bar star will inactivate the Barnase in this stage. अब इसका male infertile नहीं रहेगा because once this comes, I will introduce bar star. Bar star will now stop the activity of Barnase. Bar star has come to stop this. Now there is no longer male infertility. I can keep having self-fertilization. I will now self-fertilize this plant for 11 generations. And then finally see beneficial effects. I am sure to see beneficial effects after 11 generations. Therefore, genetically modified Dhara mustard hybrid 11. Problem of DMH11 is, it is very susceptible to weeds or herbs. क्या होता है? आपने DMH11 यहां पर ऐसे grow किया. You grew DMH11 like this. Right? ऐसे grow होगा, plant होगा, इसकी roots होंगे, some flowers, etc. Right? Many weeds come up, ऐसे weeds, and they take up the nutrients that are meant for this plant. Very susceptible to weeds of DMH11. अब मैं इतनी genetic modification कर ही रहा हूं, तो I insert one more gene at this stage. I insert one more gene. This is called bar gene. Third gene, bar gene. Bar gene क्या करता है? Bar gene makes it herbicide resistant. Resistant to herbicide. What is herbicide? Pesticide. Any chemical used to kill pests. Herbicide. Chemical used to kill herbs. This is how it was growing. Herbs were taking away, weeds were taking away its nutrients. If I were to spray a chemical here, अगर मैं पहले ही यहां पर chemical spray करता, तो these herbs would die. But this would also die. दिस वुड ऑल्सो डाई, प्लांट भी मर जाता। तो अब मुझे एक ऐसा सिस्टम चाहिए कि मैं हर्बिसाइड स्प्रे तो करूं, दीज कीप डाइंग, प्लांट डज नॉट डाई, प्लांट सर्वाइव्स, सो आई हैव टू मेक इट रेजिस्टेंट टू दिस हर्बिसाइड, कि ये वाला हर्बिसाइड यूज करूंगा, ये तो मर जाएंगे, ये नहीं मरेगा। सो आई हैव पुट 3 जीन्स inside DMH11 called Barney's, Barstar and Bar. एक बार पूरी प्रोसेस रिपीट करता हूं। DMH11 has fertilization, or it's a hybrid plant, but I also genetically modify it. Why? Because in a mustard, male and female are present on the same plant, so it generally self-fertilizes. I wanted to hybridize Varuna and Hira together to get beneficial effects. I could not do this hybridization because self-fertilization was happening. To stop self-fertilization, I first put a gene called Barnase. Barnase made it male infertile. Through male infertility, I was able to stop, through male infertility, I was able to stop self-fertilization. Then I could hybridize Varuna and Hira variety. I hybridized Varuna and Hira variety and created this. But male is still infertile. I now want, after the hybridization, I want कि यार अब ये self fertilize कर ले. For self fertilization now, I have to inactivate this Barnase gene. To inactivate this Barnase gene, I put the second gene called Barstar and I plant this. Now I will self fertilize for 11 generations and get beneficial effects. What are the beneficial effects? Yield is much more increased, 30-40% increase in yield. Okay. Problem of this was now that if I plant it in a field, herbs would arise and herbs would take away all the nutrients. If I use herbicide, plant also dies. So I have to make it herbicide resistance also. So I put the third gene called BAR. Therefore, DMH11 is a genetically modified mustard known as Dhara mustard hybrid 11. Why 11? 11 generations. It is herbicide tolerant. बात समझ आ रही है? किसने बनाया इसको? दिल्ली यूनिवर्सिटी हैज क्रिएटेड दिस. इट इज अ पब्लिकली फंडेड प्रोजेक्ट फ्रॉम DU. देयर इज अ सेंटर फ्रॉम जेनेटिक मैनिपुलेशन ऑफ क्रॉप प्लांट्स इन DU. It was created by using Barney's Barstar technology for genetic modification and by adding genes from a soil bacterium that makes mustard a self-pollinating plant. Barney's and Barstar are coming from some soil bacterium. ठीक है? Name of soil bacterium you don't have to remember, but I'll tell you just in case गलती से अगर कहीं आ गया. ये जो genes हैं कहां से उठाई हैं? Bacillus amylofaciens. This is the name of the bacteria. Don't need to remember. एक बार माइंड में बस अगर देख भी लोगे तो भी बात बन जाएगी. राइट? अब it allows for makes the mustard self pollinating plant. ये जीन इसको रोकता है. Allows for cross pollination and hybridization. DMH11 contains 3 genes. Bar gene is for making it herbicide tolerant. Barnase and Barstar, all of these are sourced from a soil bacterium called Bacillus amylofaciens. Bar gene made the plant resistant to herbicide names Basta. Basta नाम का एक herbicide है, उससे ये resistant हो गया. Is DMH11 clear? Difficult to understand, I know, but there has been a previous year question on this. It's in the news. There was an article in August in the Hindu. There was an article in July in the Hindu. There was a Supreme Court judgment in July. Therefore, it's important we have to understand it. आपको याद इतना ही करना है. अब what is the case that's happening in the Supreme Court? Google करना. I can show you also actually here. So look at this. Article in January, article in October. I'll just show you the article that shows the SC judgment. Supreme Court delivers split verdict on nod for GM mustard in July 23, 2024. So very important for us. The SC has delivered a split verdict on GM mustard. अब यहां क्या कहानी है ये बताता हूं. यहां क्या हो रहा है? There is something called, we'll read this in environment in more detail. There is something called the precautionary principle. What is precautionary principle? That if you think that something might infect the environment, that something might have a bad impact on the environment, it is better to be precautionary. It is better to take precaution rather than going for it and then seeing if it would have pollution or not. अगर आपको लग रहा है कुछ environment पे bad impact किसी चीज का होना है, we don't know. मैं एक चीज करूं उससे environment का bad impact हो भी सकता है. It's always better to be precautionary, to take precaution and not do it till you know that there is going to be no impact on the environment, rather than actually doing it कि मैं वो कर लूं, मैं DMH11, let's say DMH11 has a bad impact on the environment, it might. So it's better to have a precaution, then look at, one option is कि मैं DMH11 सीधा कर दूं plant, right? I just plant the DMH11 and that DMH11 is going to lead to various environmental impacts or not. I can see after planting. One is I remain cautious, I take precaution, I don't plant, I research further till I am sure that there is not going to be an environmental impact. I stay precautious. In environment, there is a very clear principle called the precautionary principle, agreed at the global level. अगर आपने थोड़ी भी environment पढ़ी है तो आपने सुना होगा, नहीं तो when I teach you environment, I'll teach you in detail. ठीक है? Precautionary principle है. Supreme Court के सामने ये केस गया. Now look at the problems that exist in DMH11. One, it is herbicide tolerant to a particular herbicide called basta. So one side of the, there are two sides obviously. There was the government on one side and the petitioner on the other side. The petitioner argues that, one, you are making this herbicide almost compulsory for everyone. They have to use this herbicide now. This herbicide can have a lot of environmental effects. Then you are playing with biology. This can wipe out natural varieties of mustard. तो आपने modify कर दिया है. You've played with biology. You've created a synthetic biological compound. It can, wipe out natural varieties of mustard. Thirdly, mustard oil is also consumed by humans. If mustard oil is consumed and it has these genes, what is the problem that humans will get? On one side, there is the government, which has the GEAC. GEAC हम अभी पढ़ेंगे. The GEAC's job is to regulate GM crops. So why was it a split verdict? It was a two-judge verdict. The Supreme Court में केस गया कि should we allow this or not? So there were two judges. One judge said that he is satisfied with the government's arguments, that they have done enough scientific research to allow the environmental release. The other judge said that I am not satisfied, not that the science is wrong or anything. He says that still, citing the precautionary principle, it should not be released. The main outcome of this judgment was that the government has been asked to create a new GM policy, a new policy on genetically modified crops so that these disputes don't arise. This now, what happens to this issue? It is now going to a three-judge bench headed by the Supreme Court, by the Chief Justice of the Supreme Court. It's now going to a three-judge bench. There, this matter will be decided. In a three-judge bench, a split verdict is not possible anyway, right? तो ये हुआ है, ये कहानी है of DMH11. याद आपको इतना ही करना है, एक्सप्लेनेशन बता दी. करंट अफेयर्स जब थोड़ा सा देख रहे होंगे तो दैट जजमेंट आल्सो आई शो यू. दैट्स व्हाई इट्स इन द न्यूज़. Now, what are the challenges of GM crops? Means के लिए important है. I'll just tell you in brief. One is wild species and pollution of gene pool. आपने क्या किया? आपने एक कॉटन बना दिया, which is BT cotton. BT cotton is resistant to bollworm pest. दूसरा आपने बना दिया नॉर्मल कॉटन. अब क्या होगा? If you let nature take its course, survival of the fittest will happen. Only BT cotton will survive. क्योंकि BT cotton को तो जैसे let's say there were humans, one sort of humans who would never get any infection, and second set of humans who were susceptible to all infections. After time, only one set of humans will exist because they have a natural advantage. Here also you've done the same thing. BT cotton has advantage of not getting infected by bollworms. Second cotton, natural cotton, will get infected by bollworms. Bollworms will eat it. So natural cotton बचेगा ही नहीं, right? you have eliminated the wild species. अब मान लो, अब क्या इससे प्रॉब्लम क्या है? What will happen is that there are two types of cotton. There is wild cotton and there is BT cotton. You have made a system where only BT cotton will survive, wild cotton will get eliminated because bollworm infections will happen here. अब मान लो in nature somehow some virus comes that only infects BT cotton. ऐसा वायरस आ सकता है ना? वाइल्ड को आपने खत्म कर दिया, BT cotton and it's obviously synthetically modified, it's not natural. एक वायरस आया जो सिर्फ BT cotton को इन्फेक्ट करता है. Now what do you do? Now you will lose your entire population of cotton because this you did not save. ये आप सेव करते तो there were many strains here. Some would have survived this virus. Now you have made everyone the same. थोड़ा सा एक डिजीज आ गई तो बात खत्म. Let's say you make all humans have blue eyes. Every human gets blue eyes. आपने modify कर दिया और सारे humans को blue eyes हो गई. Everyone gets blue eyes. And then somebody gets a disease or a virus comes that only infects people with blue eyes. All humans will die. On the other side, if you have natural variation, somebody has blue eyes, somebody brown, somebody black, right? अगर blue eyes वाले humans मर भी जाते हैं, humanity as a race will not cease to exist. But यहाँ आपने सब कुछ same कर दिया. You have polluted the gene pool and eliminated the wild species. Second, in GM crops there is a challenge of safety with respect to human consumption, जो हम mustard oil में देख रहे थे. Mustard oil is eaten by humans. If humans eat these barnies, bar star, bar jeans, what is the impact it's going to have on us? Cotton में problem नहीं है. I have to wear the cotton shirt. I don't have to eat cotton. But बाकी जगह तो हो सकती है problem. Then trait fees and terminator gene technology, ये दोनों समझते हैं. ठीक है? What is trait fees? Who has developed this technology of BT cotton? Obviously, this would have taken a lot of research. There is a company called Monsanto. Monsanto has developed technology called BT cotton, right? इस technology को develop किया Monsanto ने. Now, if any Indian company wants to produce BT cotton seeds, they have to pay royalty to Monsanto. We will understand why royalty when we read IPR. But they have to pay a certain amount. That increases the cost of seed production, increases the cost of input for the farmer, makes cotton costlier. ये problem तो है. Second is terminator gene technology. अब Monsanto वाले बहुत smart हैं. क्यों smart हैं? Let's say आपने आज एक BT cotton की seed ले ली. You took a seed of BT cotton plant. आप इस seed को क्या करोगे? Plant करोगे. ऐसे एक BT cotton बन जाएगा. अब plant doesn't only produce cotton. Plant खुद भी तो seeds produce करता है. It also produce seeds, right? Wheat में जो हम खाते हैं, wheat plant, उसमें जो हम खाते हैं वो seeds ही हैं. आप उन seeds को जमीन में गाड़ के give it irrigation, give it fertilizers, it will grow into a different wheat plant. अब ये BT cotton था. BT cotton is producing seeds. Farmer could have used these seeds to plant BT cotton again next year. Now Monsanto will lose out because you will buy the first set of seeds. सबसे पहले तो आपको seeds खरीदनी पड़ेंगी. First set of seeds of BT cotton you will buy from Monsanto, और Monsanto will get paid for it. इसके बाद क्या होगा? You get second set of seeds. These set of seeds farmer will plant, then third set of seeds, then fourth set of seeds. Now farmer will never go to Monsanto. तो Monsanto ने क्या किया? They introduced along with BT gene, they introduced a gene called terminator gene. What is terminator gene? ये जो second set of seeds बनेंगी, they are infertile. इनसे plant बनेगा ही नहीं. तो अब आपको क्या करना है? If you want to plant BT cotton, every time go to Monsanto and buy new seeds. They have done this so that they keep making profit and they have the right to do this. I don't think any of this is unethical. Right? उन्होंने इतनी research अगर करी है, but it creates a burden on the Indian farmer. ये unethical मेरे हिसाब से नहीं है. This is normal. Right? If they have created this technology, they are right to deserve some profit for it because so many years of research would have gone into this. तो एक बार आप प्लांट करो, आप इससे पैसे कमाओ, दोबारा आपको सीड्स चाहिए तो यहाँ जाना पड़ेगा. But then again, for the farmer, this is very problematic. He could not use his own seeds now. This is called terminator gene technology. When such plants are planted, they will produce crops only once. The resultant crop seeds will be sterile. Sterile is another word for writing infertile. Farmer is forced to buy new seeds every time. Then there is a problem of genetic divide. Big farmers will be, there is, for example, digital divide. People who have access to internet and people who don't have access to internet. There will be farmers who are rich, who can afford these seeds, who will be genetically having better plants in their fields, and there will be farmers who will not be able to afford these seeds. So it will create a genetic divide between small and large farmers. These are the challenges of GM crops. How are GM crops regulated in India? What is the regulation of GM crops in India? अब देखो ये जो पार्ट है ये प्योरली थियोरेटिकल है. Nothing for me to explain, so I will run through it quickly and I will expect that you will read it yourself. ठीक है? Because देखो यार जो भी अभी तक scientific part है, I am explaining it in detail. This I will expect that you can read yourself. Is that okay? Because if I spend time here, then explanations will become, because we have limited time, there is nothing to explain here. I'll tell you whatever is to explain. I will expect that you will read this yourself. है ना? ऐसा नहीं है मैं आपको बिल्कुल नहीं पढ़ा रहा हूं, बट आई विल एक्सपेक्ट कि आप खुद से एक बार पढ़ोगे. देखो ब्रॉडली आपको बता देता हूं. ब्रॉडली द रेगुलेशन ऑफ जीएम क्रॉप्स, इट हैपेंस बाय द जीईएसी एंड देन द एनवायरनमेंट मिनिस्ट्री. कैसे? लेट्स सी. देखो सबसे पहले वो जो, अगर आपको जीएम पे कोई मेंस का क्वेश्चन आ जाए, राइट, वी हैव सीन द एडवांटेजेस ऑफ जीएम क्रॉप्स, then we have seen the challenges of GM crops, we have seen the examples of GM crops. Now let's see what the economic survey of 2017 said with regards to GM crops. Mains ke liye hai. What did they say? One, allow GM crops for disease and pest control. Allow GM crops that give us a long shelf life. What is long shelf life? You create a tomato today, you put it on the shelf. If it's on the shelf, it will rot in 20 to 30 days. 20-30 days में वो uneatable हो जाएगा. If you have to export this tomato, it will take 50-60 days to export, maybe 30 days to export. Wahan jaake it needs to again stay on the shelf for some time for someone to buy it. So it needs to not rot for 50 to 60 days if it has to be exported efficiently. So it can stay on the shelf for a long time. It should not rot in on the shelf. Shelf पे खराब ना हो जाए पड़ा पड़ा. Long shelf life जो जीन्स अलाउ करती हैं उनको अलाउ कर दो. शॉर्ट क्रॉप ड्यूरेशन, ऐसे क्रॉप्स जो आज मैंने प्लांट किए विद इन 2-3 मंथ्स आई कैन हार्वेस्ट, उनको अलाउ करो. एंड नॉन फूड को अलाउ कर दो. दैट इज व्हाई बीटी कॉटन इज अलाउड. इवन इफ देयर इज अ चैलेंज बाय इंजेस्टिंग बीटी जीन्स, इट माइट क्रिएट अ प्रॉब्लम इन माय बॉडी. आई एम नॉट गोइंग टू ईट कॉटन, सो कॉटन में आप अलाउ कर सकते हो. दे सेड दैट यू अलाउ जीएम सीड्स व्हिच आर लो कॉस्ट एंड विदाउट टर्मिनेटर जीन्स एंड एनकरेज domestic production of GM crops so that there is ending of monopoly. Why am I telling you this in detail? Because of the judgment of the Supreme Court last year. You will either get asked this in the mains or in the prelims, so I am covering both. हमने prelims के लिए सारी technologies समझ ली, mains के लिए हमारे पास अच्छा conclusion है. We know the advantages, we know the challenges, we'll be able to write. अब these are, I'll come back to this. These are applications of biotech. I will come back to this. We'll read these in detail. अभी इसको छोड़ते हैं. Right? Let's look at first the regulation of GM crops in detail, then we'll come back to those applications. So regulation is, one is GEAC, Genetic Engineering Appraisal Committee. Then it is approved by the Ministry of Environment, Forest and Climate Change. In case of DMH11, there is still vacillation. Vacillation means going back and forth, allowed, not allowed, Supreme Court judgment, ye wo. So there's still no clear, idea whether it's allowed or not. Environmental release has been allowed, commercial release has not been allowed. Again, it's in the Supreme Court. I doubt you will be asked a question on its current status because it's amenable to change at any time. Right? BT brinjal, अब same जैसे BT cotton, BT brinjal was introduced. BT brinjal. Problem क्या हुई? Lot of side effects. 33 side effects were seen in BT brinjal, so it was banned. Introduce किया, try किया, ban कर दिया. Then new issue with BT cotton. अब आपने BT cotton allow किया हुआ है. There is a new pest called white fly. White fly can infect BT cotton. Huge problem. That is why BT cotton also is facing problem. There is also issue of pest resistance. Let's understand this. Very interesting. ये है BT cotton. Let's say these are 5 bollworms. Bollworm 1, 2, 3, 4, 5. Bollworm 1 will eat this, it will die. Bollworm 2 will eat this, it will die. Bollworm 3 will eat this, it will die. Why? Because there is a protoxin that is getting released and it is killing them. Now, by some gift of nature or due to some mutation, bollworm number 5 has a gene that makes it resistant to this protoxin. It does not die because of the protoxin. ऐसी हो सकती है ना कोई जीन इसके अंदर प्रेजेंट हो. अब देखो क्या हुआ. These 4 bollworms died, but this bollworm number 5 did not die and ate the entire ball. Now what happens? The entire population of bollworms, over time, only these type of bollworms will survive who have this gene. But now if अब ये इस टाइप के सारे मर जाएंगे, let's say there are 10 crore bollworms, only 1 crore will be left who have this gene. But now these 1 crore will produce 2, then 4, then 8. There will come a time when all bollworms have this gene. Now your BT cotton is ineffective. The pests have developed resistance. Problem of pest resistance. Right? Up. For a conclusion, you can take the statement that Shri M.S. Swaminathan, I'm sure you know who this is. नहीं पता है तो please Google करना. You should know who this is. He called BT crops a failure and government should resort to genetic engineering only as a last resort. Otherwise, BT crops are a failure. That's what he said. So you can use this in the conclusion also. Now let's look at the regulation behind GM crops. Firstly, there is a global regulation. There are 3 protocols, Nagoya, Cartagena and Nagoya Kuala Lumpur Protocol. We will study these with the environment. These are protocols to the Convention on Biological Diversity. There was the Rio Summit in 1992. 3 conventions came out of it. The Convention to Combat Climate Change, the Convention to Combat Desertification, and the Convention on Biological Diversity. These are the 3 protocols of the Convention of Biological Diversity. Environment में पढ़ेंगे. अभी just read it. Don't remember it. I will explain each of these in a lot of detail. अब India में this I want you to read by yourself. I'll give you a summary. Right? Basically India में क्या है? ये वाला part, ये वाली slides में चाहूंगा आप खुद से पढ़ो. There is a regulatory framework. There are 1989 rules. These rules are made under the Environment Protection Act. ठीक है, ये सारी चीजें कवर करते हैं. Sale, storage, export of all GM crops. ठीक है. There are 6 committees. सबको जितने लोग यूपीएससी के लिए प्रिपेयर कर रहे हैं, जितनों को मैं जानता हूं एटलीस्ट, they only know name of this committee. But this is not how GM crops are regulated. There are 6 committees. ठीक है. There is an RDAC. RDAC का advisory job है. Then there are 3 other committees, जिनमें से एक GEAC है. Then there are 2 committees at the ground level. ये advisory committee है. इसका क्या काम है? Advise करना गवर्नमेंट को कि रेगुलेशन कैसी होनी चाहिए. फिर अप्रूवल लेने के लिए देयर आर 3 टाइप्स ऑफ कमिटीज. फिर अप्रूवल तो मिल गया, अब आप प्लांट करोगे एट द डिस्ट्रिक्ट लेवल. दिस आल्सो हैज टू बी मॉनिटर्ड दैट योर प्लांटिंग हैज बीन डन प्रॉपर्ली और नॉट. सो देयर इज अ डिस्ट्रिक्ट लेवल कमिटी एंड अ स्टेट लेवल कमिटी फॉर मॉनिटरिंग. द हेड ऑफ दिस डिस्ट्रिक्ट लेवल कमिटी इज द डिस्ट्रिक्ट मजिस्ट्रेट हु इज एन IAS, and that is why your knowledge of biotechnology is important. If you don't know any of this, right, you will not be able to head this committee properly. आप चला ही नहीं पाओगे इस कमिटी को. right? अब here the names of all the committees are written. RDAC का काम, Institutional Biosafety Committee का काम, Review Committee on Genetic Modification RCGM का काम, GEAC का काम, SBCC का काम, DLC का काम. This I want you, these 2 slides I want you to read by yourself. समझ पढ़ के समझ आ जाएगा. right? Just the job of these committees is important. अब most important is GEAC. वो क्या क्या काम करती है? I want you to read this by yourself also. And what it functions under. जैसे GEAC is functioning under MOEFCC. And since there is committee in the name, 99.9% if there is committee in the name, it is headed by some civil servant. If there is council in the name, it is generally headed by some minister. So committee, civil servant, council, minister, this is not a universal rule, right? But this is mostly the rule. So let's say GEAC, 24 members, who is it chaired by? A special secretary or an additional secretary of the ministry. So obviously this person would be an IAS officer or forest service officer. So obviously a civil servant is heading this committee, but I want you to read this by yourself. This I want you to read by yourself. This I'll explain to you. I will explain this to you, but I want at least once you should read by yourself. At least once you should read this by yourself. ठीक है? This also I want you to read by yourself. ये क्या है? There are various ministries. It is not only MOEFCC that is responsible for control of GM crops. There are various other ministries involved. हर मिनिस्ट्री क्या काम करती है? देखो याद नहीं करना है इसको. It is very logically done. जैसे यहां पर मिनिस्ट्री ऑफ हेल्थ है. अब मिनिस्ट्री ऑफ हेल्थ क्या कर रही होगी जीएम क्रॉप्स में? मिनिस्ट्री ऑफ हेल्थ वुड बी सेइंग कि ह्यूमन हेल्थ पे अगर कोई इंपैक्ट आएगा तो हम रेगुलेट करेंगे, राइट? देयर इज मिनिस्ट्री ऑफ कॉमर्स. मिनिस्ट्री ऑफ कॉमर्स क्या कर रही होगी? दैट इफ यू आर ट्रेडिंग विद अदर कंट्रीज तो वो पार्ट हम रेगुलेट करेंगे. जस्ट रीड दीज स्लाइड्स. आई वांट यू दीज 5-6 स्लाइड्स आई वांट यू टू रीड बाय योरसेल्फ. ये स्लाइड की अभी क्या स्टेटस है? नॉर्मली क्या होता है, जो जीईएसी है, जेनेटिक इंजीनियरिंग अप्रेजल कमिटी, दिस गिव्स परमिशन फॉर एनवायरमेंटल रिलीज, कि एनवायरमेंट में रिलीज कर दो इन क्रॉप्स को. देन यू टेस्ट इन द एनवायरमेंट. देन देयर इज फाइनली commercial release, that you can use these commercially. You can use these commercially for growing by farmers. तो अब BT cotton approved by GEAC, approved by ministry, allowed commercially. BT brinjal banned by ministry. GM mustard, GEAC approved, ministry rejected, went to Supreme Court, still in the Supreme Court. GM rubber trials are going on and GM soy meal import has been allowed. Again, these slides on GM soy meal. What is GM soy meal? What is GM rubber? I want you to read by yourself. एक बार पढ़ लो. कल की जो क्लास होगी उसमें मैं आपको जल्दी से एक्सप्लेन कर दूंगा. But I think if you have read by yourself once, it will become very easy. कुछ है नहीं यहाँ समझने के लिए, right? But it saves a lot of time for us, and you know the concept behind it. It's not a big deal. So I think these 10 slides. Yeah, this is probably the last one. Yeah, and this one. ये मैं आपको कल पढ़ाऊंगा. This will take time. तो यहाँ तक, राइट? यहां तक दिस विल रीड अभी. सो आई वॉन्ट यू टू रीड फ्रॉम, आई जस्ट, ये स्लाइड्स अपलोडेड हैं, आई जस्ट टेल यू फ्रॉम, देयर आर टोटल 10 स्लाइड्स दैट यू हैव टू रीड फ्रॉम दिस जीएम क्रॉप्स इन इंडिया. ये ग्लोबल वाला तो हमें पढ़ना नहीं है, ये तो मैं आपको पढ़ाऊंगा एनवायरमेंट में. फ्रॉम दिस स्लाइड जीएम क्रॉप्स इन इंडिया टिल दिस स्लाइड GM plants. These are total 12 slides. ये 12 slides just read once. इसमें जो भी important है हम कल डिस्कस कर लेंगे, but if you read once, my job becomes very easy and I can focus on the explanation. ठीक है, इनको एक बार रीड कर लेना. अब let's come back. We skipped the applications of GM, तो वो देख लेते हैं. यहां तक हमने देख लिया था. Now we'll see other applications of biotech. तो अभी तक हमने क्या पढ़ा है? We have read GM crops in a lot of detail. Now we will read forensic biotechnology, which is DNA fingerprinting. After DNA fingerprinting, we will read something called transgenic animals. GM animals. अभी तक हम GM plants पढ़ रहे थे, फिर हम GM animals पढ़ेंगे. After we read transgenic animals, I want to teach you a system today called CRISPR-Cas9, which is very interesting. Also, the Nobel Prize was given for it. Also, I think 3 times UPSC has asked question from CRISPR-Cas9. ठीक है? Let's take a small 2-minute break. आपको I hope समझ आ गया. You've understood what you have to read by yourself. एक बार read करके आना है, not skipping anything, but at least read it once. है ना? समझ आ गया? खुद क्या read करना है? Those 10 slides you have to read. है ना? एक बार रीड कर लेना. आई विल एक्सप्लेन वॉट एवर इज इंपॉर्टेंट देयर, आई विल एक्सप्लेन. आई जस्ट वांट यू टू रीड इट वंस. ठीक है? ओके. अब वी विल रीड एप्लीकेशंस ऑफ आरडीटी. अब ये देखो, अरविंद ने क्वेश्चन किया है वायरस. देखो, आई टेल यू व्हाट, मे बी यू आर क्वेश्चनिंग दिस विद द बेस्ट ऑफ इंटेंशंस. but I get slightly triggered when I see messages like this, and I'll tell you why. ऐसा हो सकता है मैं आपको वायरस ना पढ़ाऊं, नहीं हो सकता ना. But when you write this, maybe it's not your intention, maybe it's completely my thing. I sometimes feel when this is written that you are not trusting that I will teach you everything. Okay? I know that might not be your intention. That is sometimes only the feeling that gets conveyed to me. So please don't write things like this. This is just a request. I know you don't have any bad intentions. गलती मेरी ही है, आपकी नहीं है. Probably you did not want to say anything like this. You just asked when we'll read virus. But मैं इससे थोड़ा I get impacted. So I'm just requesting if you would not write this. ऐसा नहीं हो सकता ना कि मैं आपको वायरस ना पढ़ाऊं. Obviously यार मैंने खुद बोला है 4 बार कि हम वायरस पढ़ेंगे. So how is it possible that I don't teach you virus? I will teach you viruses. It's just I'm trying to make it. I'm going to teach you in the best possible sequence. These are very interrelated concepts. अगर आप NCERT से भी पढ़ोगे उसमें भी 11th class में कुछ है, 12th में कुछ है. I am trying to create a best sequence for you to understand everything in one go. Because, for example, when you have to understand gene therapy, which is also something that we'll study today, you need to have virus context. For viruses, you need another context, right? So, थोड़ी problem है. There is some disorganization, and I think I told you this even in the first class, that you'll understand everything about GM or genetic modification and biotechnology only when all our classes are complete, maybe after 5 classes. This is our 5th class. I think by tomorrow you'll understand everything put together. But वो थोड़ी sequence का issue इसमें है. This just the subject is such that issue does arise, right? Anyway, so I know you're just asking when we'll do it, and we'll definitely do it. Just please don't write messages like this. This is just a request. Anyway, so we'll take a break for like 4 minutes, and then we'll continue with applications of biotech from here. These are just the applications. We'll just read them. The real application we'll read now is 1, DNA fingerprinting. 2, then we'll read transgenic animals. Then probably we'll read gene therapy. I'll give you a brief on viruses first, and then we'll read gene therapy. Then we'll read immunity, and if possible CRISPR-Cas9 today only, right? थोड़ा सा एक्सटेंड होगी क्लास, पहले ही बता रहा हूँ. 12:30 छूटोगे आप. ब्रेक ले लो, शांत 4 मिनट आराम से बैठो, हैव सम वाटर, look away from the screen, right, for the next 4 minutes, but it will take time today. Okay, we come back after 4 minutes. All right, let's continue. So, देख लो. Whenever you get a question on applications on biotech, and you will get it because every alternate year either biotech comes or nanotech comes, right? देखो अब applications of biotech again, if I ask you to write, a lot of people will be able to write, है ना? अब हम और आगे पढ़ेंगे, we'll read various other applications. Everybody will be able to write, and you will not be able to differentiate your answer from your competitors. So the one good way to differentiate your answer from your competitors is to use this. Now, what is this? This is basically branches of biotech, right? So these are various branches of biotech that you can use, and they are color based. So these are applications of green biotech. Green biotech is using biotech for agriculture. So for increasing yield of plants, for having biopesticides, example is BT brinjal, BT cotton. For having high yielding varieties, for example GM mustard. For doing biofortification. What is biofortification? We add some nutrients. We'll read biofortification in detail. Adding some nutrients to various crops. For example, we saw golden rice. Rice में विटामिन ए नहीं होता, but आप विटामिन ए की जीन्स डाल दो राइस में. Rice will start expressing vitamin A, and vitamin A will then be consumed with rice. Up. I want you to think about a question. There is one case where I can give vitamin A की deficiency is very common. Vitamin A के supplements दे दूं मैं. One option is I give supplements of vitamin A to everyone in the population. ठीक है? कि मैंने सबको कहा कि मैं सबको फ्री में supplements बांटूंगा. You eat that supplement, you eat that tablet, you will get vitamin A. Second case is I have put vitamin A inside rice. Let's say the cost of both of these, cost of the first mechanism and the second mechanism, the cost is the same. Cost to the government, cost to the consumers is the same. Which of these is now better according to you and why? Think about it. ये दोनों options हैं. I can give vitamin A supplements, or I can put vitamin A in rice and then give it. Which of these would you say is better and why? सोच के बताओ. I'm turning the chat on. Which of these would you think is a better option? One or two? Yaar, why? I just want to know why. Rice mat likho. Good. Vagish, best answer so far? Right. So, if you put it in rice, you are not asking people to change their behavior. Their behavior stays the same. They just have to keep eating rice. अभी भी तो rice खा रहे हैं, बाद में भी rice खाते रहेंगे. India is facing a huge problem of, you must have heard, hidden hunger. What is hidden hunger? देखो, there are some macronutrients that are required. Macro से नाम से clear है. Macro means big, required in large quantities, like carbohydrates, proteins, fats, required in large quantities by humans. There we have been largely able to eliminate hunger in the country. But there is a problem of micronutrient hunger. On the face of it, hunger is not visible. It's hidden because people are not getting enough vitamins and minerals. Micronutrient hunger is called. Micronutrient means nutrients that are required in small quantities, and micronutrient hunger would be referred to as hidden hunger. You must have heard this keyword. India has a problem of hidden hunger. So this can be a very good solution. People are eating rice a lot in India. Just let them eat rice, put vitamin A inside rice. Now there are safety concerns here because you will genetically modify the rice. That might create a problem, right? So you cannot say either one is better or two is better. Two has the advantage that there is no behavioral change, but one has the advantage that there are no safety concerns. You don't need to do any genetic modification, right? So both these things happen together. Like you should know the advantages, disadvantages of both. So biofortification, then we read you can increase the shelf life. If you increase shelf life, you will increase export ability. One sort of place where this has been done is called flower sour tomato. This is one of the first GM crops that was created. It's not there in India yet. India has not allowed it yet. Flower sour tomato has shelf life of 90 days. Normal tomato has shelf life of 30 days. Then you can deal with disease. One of the place where this was done was Samba Masuri rice. rice where this biotechnology was used, disease was not happening. Then RNA interference and plant vaccination we have seen in the case of tobacco and idea of MI melodyne incognitia infecting those tobacco plants. Then cold tolerant strawberries. Strawberries generally do not tolerate cold when being grown. You can make cold tolerant strawberries. You can make better breeds in cows, etc. So these are applications in agriculture known as green biotechnology. आपको question आए तो separate it into green. Second thing you should write is blue biotechnology. Blue biotechnology is for marine applications. So aquaculture. For seafood, you can use biotechnology. You can make transgenic fish. What are transgenic animals? We'll just see. You can make transgenic fish. Right? Transgenic fish, you can put some genes inside fish that makes them freeze resistant. Right? क्यों? अब अगर आप frozen area से fish transport कर रहे हो, they get frozen there, they die there, and then the taste is bad, they are rotten a little bit. This creates a problem. So Japan में fish eating is a huge thing that they do. So culture has a lot of fish eating. You might know sushi, for example, is a very common dish that the Japanese consume. But they all wanted fresh fish. And around Japanese waters, see, this is how you link things. Around Japanese waters, there is a cold and hot current mixing, where there is a very good area for fishing. Now, try to remember the name of that cold and hot current. जैसे ही आप ये चीजें सुनो, आपके दिमाग में रिलेशन आ जाना चाहिए. So that cold and hot current, I will not tell you the name. There are two currents. You will have to remember it. There is a cold current. The cold current starts with O. The hot current starts with K. These currents mix, right? And it creates an area of fishing. The problem is the waters around this area are cold. So if they were fishing in this area and getting these waters to, and getting these fish to Japan, but while you were transporting these fish, right, they used to get frozen, and then the taste was not as good. So क्या किया लोगों ने? Very ingenious solution. They used to fish on certain boats, and this water is not directly very close to Japan. So let's say these are the Japanese islands. They're fishing here. Now in this transport, the fish would die and get frozen, and the taste would not be good. So they would take fish, they would do the fishing, right? So जैसे इस water से आपने यहाँ पर आपने fishing करी, you put it in a boat. इस boat में क्या किया? इस boat में भी एक pool बना दिया of water. Used to put all the fish inside this pool. Fish would normally die in this pool. They would just stay stagnant and die. They put one shark inside this pool. So fish would constantly keep moving inside this pool because they now had to protect themselves against the shark. So they would remain fresh till here. Then they came here, they first killed the shark, then they took the fish. Right? Very ingenious solution by the Japanese to keep their fish fresh. The other thing you could do is, when you are doing aquaculture, when you are growing fish artificially only for the purpose of fishing them, you can put this gene inside them to make them freeze resistant. You can use this for conservation of species also. Right? You can use this for seaweed cultivation also. आपको you have to cultivate seaweeds in a specific culture. You can use biotechnology there. Biotechnology is not just genetic editing, by the way. Even traditional hybridization is biotechnology. Remember that. This is red biotech. Red biotech is for health. I want you to read all of these. I will explain all of these to you. You will understand all of these terms once we're done with biotech and health. But red biotech, any question that you ever have to write on health, there are only three things that are done in health. One is prevent the disease from happening. So in prevention में क्या क्या करते हैं? बायोटेक में vaccines, transgenic plants, transgenic animals. We've read transgenic plants, GM plants. अभी आप transgenesis और GM को same मान लो. I'll tell you the difference. Transgenic plants, GM plants give us better nutrition. Transgenic animals क्यों use करते हैं आज ही देखेंगे. This is for prevention of disease. आप disease या तो prevent कर लो. You are unable to prevent the disease. What is the second step? You diagnose it quickly. Then there are some things for diagnosing. ये हम सारे पढ़ेंगे. इसमें से एक PCR है, polymerase chain reaction. This we have already read. Other things, ELISA, DNA probes, monoclonal antibodies. This we know. And lastly, there is treatment. So either prevent. If you can't prevent, diagnose early and treat efficiently. Right? This is the whole idea of health. So prevention में ये, diagnosis में ये, treatment में we already know human insulin. Right? Human insulin हमने देखा है. Gene therapy we'll see. stem cell therapy we'll see, recombinant proteins we'll see. But just to give you an idea, हम सारी चीजें ये देखने वाले हैं. Then there is environmental biotechnology. Environmental biotechnology में firstly there is something called bioremediation. अब again word breakdown. आपने सुना होगा remedial classes. Remedial classes क्यों होती हैं? आपको कोई चीज एक बारी में समझ नहीं आई, to restore the condition, to restore your doubts, to get your knowledge back to the original state, we do remedial classes. And you missed out on something, कुछ रह गया था, उसको ठीक करने के लिए. Remediation means correcting this or correcting a deficit. Remedial classes to correct the deficit. Bioremediation, using biology to correct some deficit, correct some defect. What are we doing here? We have made something called oil zapper bacteria. Means पे question आया हुआ है इसमें. Oil zapper bacteria are, example is Oleovorus S, made by TERI. TERI is the Energy Resource Institute of India. I think that's what it's called. I'm forgetting the full form, but TERI is very famous. It's the body that makes Tetra packs, which all our juices, etc. come in. So TERI made Oleovorus S. This is a bacteria that can be used for bioremediation. How? जहां भी oil spill होता है, if there is an oil spill, you take this bacteria and put it there, it will eat up this oil. Oil zapper, made using biotechnology, a bacteria that would take up all this oil. Bioremediation, correcting the problem in the environment through bacteria and use of biotechnology. Bioremediation using Oleovorus S. Then phytoremediation. Remediation तो पता है, phyto meaning plants. अब कहीं पे soil pollute हो गई, right? Soil pollute हो गई, soil में बहुत सारे toxic elements आ गए. There are many toxic elements inside the soil. What if some plants could grow in this soil and take up all these toxic elements in themselves? इसके लिए हम eucalyptus plant use करते हैं. Using plants, phyto, to correct the defect in the environment, remediation. Defect क्या था? Toxic elements हैं. यहाँ पर toxic element है, आप plant ले लो. Plant will take up all these toxic elements. Eucalyptus is such a thing. So use of plants and some soil microbes to reduce toxic elements in the soil. Eucalyptus is used here. Phytoremediation. Last, DNA barcoding. DNA barcoding हमने देखा है क्या होता है. Now let's see it in detail. इस पे question भी आया है. DNA barcoding में क्या होता है कि let's say you are a forest service officer and in your area, let's say you are meant to govern this particular forest. आपको इस forest को govern करना है. You know that there are, let's say, 200 species that are present in this forest. 200 species are there, various plants and animals. आप सबका DNA लो and put it in a barcode, store it in a barcode. ठीक है, सबका DNA लो और उसे barcode में store कर दो. Now let's say you find some living material lying there. ऐसा आपको इस forest में कहीं पे कोई living material पड़ा हुआ मिला. You want to know whether this is from a new species or is it part of these 200 species. तो आप क्या करोगे? You will take this DNA. You will compare this DNA from these barcodes. If it matches, you will know कि किस species का है. If it does not match, this is a new species and you will investigate further, known as DNA barcoding. अब पढ़ते हैं forensic biotechnology and DNA fingerprinting. What are fingerprints? Why are fingerprints used? Fingerprints are different in almost all humans. Why I am saying almost all will also become clear to you. Fingerprints are different. Let's say for now, fingerprints are different in all humans. If my fingerprints are found on a crime scene, I am very likely to have been involved in a crime. I might not have done the crime, but I was there. If my fingerprints are found somewhere, you can say that हाँ ये बंदा यहाँ पर आया है, this person was there, right? We are using something called DNA fingerprinting. We are using DNA to identify whether someone is there or not. DNA fingerprinting. कैसे होता है ये? देखो, we use something here called variable number of tandem repeats. इसको पढ़ेंगे पहले. Let's understand. We'll read all of this. पहले let's understand something else. If I write now something on the board in front of you by the name of human जीनोम प्रोजेक्ट, ह्यूमन जीनोम प्रोजेक्ट, व्हाट डू यू अंडरस्टैंड? क्या कर रहा होगा ये प्रोजेक्ट? व्हाट वुड दिस प्रोजेक्ट बी डूइंग? गुड श्रेयांश। नहीं, अगर आप कलेक्टिंग जीन लिख रहे हो तो गलत है ये। ऑल जीन्स रॉन्ग। देखो मैं लिखता हूं, चलो आई विल राइट वर्सेस, आई विल राइट ह्यूमन जीन प्रोजेक्ट। ह्यूमन जीनोम प्रोजेक्ट वर्सेस ह्यूमन जीन प्रोजेक्ट। सॉरी फॉर दी हैंडराइटिंग मिसमैच। अब बताओ क्या डिफरेंस होगा? अरे अपनी प्रीवियस नॉलेज मत यूज करो, जो पता है उससे बताओ। व्हाट इज जीनोम? Genome is full complement of DNA. Let's say human DNA is like this, something like this, right? Double helix. Anyway, let me make it like this: double helix model of DNA. This is human DNA. There are some genes on it. These are genes. ये genes हैं. Human Genome Project would be studying entirety of this DNA ना, पूरा DNA पढ़ रहा होगा, complete DNA. जीन्स मिल गई बहुत अच्छी बात है, बट उसका काम जीन पढ़ना नहीं है, उसका काम पूरा पढ़ना है. We will read HGP in detail tomorrow, but reading the entire genome is the job of Human Genome Project. Human Gene Project का क्या काम होगा? अगर ऐसा कोई, none of no project by this name exists by the way, but its job would be to look at these genes. ये काम होगा Human Gene Project का, है ना? Human Genome Project is a real thing. What did Human Genome Project do? Human Genome Project was an attempt to look at entire DNA of humans. Humans का जितना भी DNA है सारा देखेंगे, पूरा DNA humans का मैप करेंगे. So what did they see? They saw that there were 3 into 10 raised to power 9 base pairs in human DNA. This is 3 billion base pairs. What are base pairs? A के सामने T, G के सामने C. This is one pair. तो इन्होंने देखा कि ह्यूमंस में 3 बिलियन बेस पेयर्स हैं डीएनए में. दिस इज व्हाट दे फाउंड आउट. दे ऑल्सो फाउंड आउट, देखो अब ह्यूमन डीएनए फॉर ऑल ऑफ अस विल बी द सेम मोस्ट ऑफ द टाइम, राइट? ऑलमोस्ट सारी चीजें सेम है, थोड़ी सी वेरिएशन है, राइट? देयर इज ओनली वेरी स्मॉल वेरिएशन. So everybody has 2 eyes, 1 nose, 1 mouth, 2 ears, hair on the head, etc., etc. There is just some small variation. So it was found that 99.9% of DNA in humans is the same. Only 0.1% DNA is different. ये ह्यूमन जीनोम प्रोजेक्ट को पता चला. Now इससे काफी सारी चीजें भी निकली. इसका हैज एन इम्पैक्ट इवन ऑन सेइंग दैट रेसिज्म, जो आपने आइडिया सुना है कि ब्लैक रेस वर्सेस व्हाइट रेस एक्सेट्रा, दिस डिस्प्रूव्ड रेसिज्म कंप्लीटली. इफ दिस इज द फैक्ट कि 99.9% डीएनए लोगों का सेम है, this much variation in DNA is not enough to produce races. So in the UNESCO statement of races you will read there are no true races in humans. This is just an idea. Indian race, humans में races होती ही नहीं है, इतनी कम DNA variation में possible ही नहीं है. Anyway, it's not important for anthropology, not important for us. But 0.1% of DNA was found to be different. अब देखो, now let's say you found 2 DNA at a crime scene. आप एक क्राइम सीन पे गए, ये एक डीएनए है, या चलो आपको एक डीएनए मिला विक्टिम का तो हटा देते हैं. दिस वाज प्रोबेबली द डीएनए ऑफ क्रिमिनल. This is criminal DNA. There are two suspects. Let's say suspect 1, suspect 2. Suspect 1 का आपने DNA कलेक्ट किया, suspect 2 का आपको DNA कलेक्ट किया. What will you have to compare here? How many base pairs will you have to compare? बताना. How many base pairs will you have to compare? सोच के बताओ. CSAT का क्वेश्चन है, गलत मत करना, फेल हो जाओगे CSAT में नहीं तो. How many base pairs you will have to compare? क्वेश्चन तो सुन लो. How many base pairs will you have to compare? How many, not percentage. जल्दी जवाब देना है सबको, है ना. एक ढंग का जवाब नहीं आया अभी तक, एक भी नहीं आया. सुनते ही नहीं हो यार तुम क्वेश्चन. दिस इज नॉट ओके यार. जल्दी लिखने क्या क्यों है तुम्हें जल्दी लिखने की. सुन तो लो क्वेश्चन क्या है. हाउ मेनी बेस पेयर्स विल यू हैव टू कंपेयर. एक आंसर ठीक नहीं है अभी तक, एक भी. हाउ मेनी बेस पेयर्स विल यू हैव टू कंपेयर. दोबारा, हाउ मेनी बेस पेयर्स विल यू हैव टू कंपेयर. अब गलत मत करना प्लीज. I don't know how you guys are thinking. Maybe I have not asked the question properly. I still haven't seen the right answer. मैं बता ही देता हूं. But I'm fairly disappointed. Let me tell you. देखो, इस DNA में कितने बेस पेयर होंगे? 3 into 10 raised to power 9 would be there. इसमें भी 3 into 10 raised to power 9 होंगे. इसमें भी 3 into 10 raised to power 9 होंगे, है ना. आप ह्यूमन जीनोम प्रोजेक्ट कर चुके हो, आपको पता है 99.9% डीएनए सेम है, 0.1% डीएनए डिफरेंट है. तो यू नो विच पार्ट ऑफ डीएनए सेम इन ऑल ह्यूमंस, राइट. एक ठीक आंसर देख लिया मैंने, चलो थैंक गॉड. यू नो विच पार्ट इज द सेम. यू नो दैट इन एवरी पर्सन्स डीएनए, इन अ ह्यूमंस डीएनए देयर इज 99.9% सेम पार्ट, 0.1% डिफरेंट पार्ट. आपको पता है कौन सा पार्ट सेम है, तो जो पार्ट सेम है आप उसको थोड़ी कंपेयर करोगे. व्हाई वुड यू वांट टू कंपेयर द सेम पार्ट. तो यू नो देयर इज ओनली 0.1 डिफरेंट पार्ट. हर डीएनए में ओनली 0.1% इज डिफरेंट. व्हाट इज 0.1% ऑफ 3 into 10 raised to power 9? इट इज 3 into 10 raised to power 6. तो आप इसमें से जो वो 0.1% पार्ट है जो डिफरेंट है वो निकाल लोगे. 3 into 10 raised to power 6 इसके निकाल लोगे, 3 into 10 raised to power 6 इनके निकाल लोगे, 3 into 10 raised to power 6 इनके निकाल लोगे. देन यू विल कंपेयर कि यार इससे ये मैच कर रहा है या ये मैच कर रहा है. अब समझ आया. अब समझ आया क्या बोल रहा था मैं. ठीक है. अब आप सोचो 3 into 10 raised to power 6 is how much? 3 with 6 zeros. ये आंसर भी कई लोगों ने दिया था, 30 लाख बेस पेयर्स. ठीक है. अब आप मुझे बताओ, अब आप क्या करो इसमें. विल यू रीड 30 लाख बेस पेयर्स फॉर एवरीबडी. यहां पर ऐसे सीक्वेंसेस लिखी हुई हैं, दिस इज 1 बेस पेयर. ऐसे आप सीक्वेंसेस पढ़ोगे क्या 30 लाख. सो दिस वाज नॉट पॉसिबल. दिस वाज नॉट पॉसिबल. आप यू कैन नॉट रीड 30 लाख सीक्वेंसेस. इवन इन अ कंप्यूटर इट्स ऑलमोस्ट इम्पॉसिबल टू रीड. तो ये तो नहीं हो सकता, ये काम तो नहीं हो सकता. बट दिस मेथोडोलॉजी इज साउंड. If there was a way to compare these many base pairs, this methodology was sound and obviously very helpful in criminal investigation. तो अब इसको समझते हैं. After Human Genome Project, they saw that 99.9% DNA is the same in all humans. This was called bulk DNA. 0.1% DNA is different, called satellite DNA. So satellite DNA and bulk DNA. This is what it was called. That in all. अब ये प्रॉब्लम हमें सॉल्व करनी है, कैसे करेंगे वो प्रॉब्लम सॉल्व करते हैं, राइट? सो satellite DNA and bulk DNA, 99.9% DNA is same, called bulk DNA. 0.1 is satellite. Why was this called bulk and satellite? Because जब आप इसको centrifuge करते हो, you have to separate this bulk and satellite now. When you centrifuge it, bulk DNA collects in one place like this, and satellites collect at the sides. So bulk and satellite. ठीक है, not important but just the scientific process. अब आपको satellite DNA compare करना है. Obviously, this satellite DNA will have differences. This is the part that is different, right? अब इस satellite में भी coding non-coding part दोनों होंगे. I hope you are able to understand. In humans, between me and you, there are some differences in coding part, more differences in non-coding part. बात समझ आ रही है? Between me and you, there are differences in coding part also, because we look different, we speak different, our eyes look different, our hair looks different. There are differences in coding part. There are also differences in non-coding part. All the differences, coding plus non-coding differences, are here in satellite DNA. समझ आ रही है बात? Non-coding में differences ज्यादा होते हैं. Non-coding has more differences, right? But that does not mean that satellite DNA is non-coding. ठीक है, अच्छा. This differences in DNA, differences in humans, is called, इसको बोलते हैं हम polymorphism. Again, word breakdown. Poly means many. Morph means shapes. A literal word would mean many shapes, but many types. किसी का satellite DNA कैसा है, किसी का satellite DNA कैसा है. Differences, many shapes are there. Polymorphism is there. In this satellite DNA, there is a lot of polymorphism. In this satellite DNA, there are a lot of polymorphism. There are a lot of differences. Everybody has differences. But there are 30 lakh base pairs here. I cannot compare 30 lakhs. तो क्या देखा गया? That in the non-coding part in this satellite DNA. तो ये देखो, I'll just write it like this. ये जो satellite DNA है, it can be divided into itself coding and non-coding part. कुछ थोड़े बहुत कोडिंग डिफरेंसेस यहां पे होंगे, बहुत कम होंगे, वेरी रेयर, ऑलमोस्ट नॉन, वेरी स्मॉल डिफरेंसेस वुड बी देयर. कुछ यहां पे नॉन कोडिंग पार्ट होगा जिसमें डिफरेंसेस होंगे. बोथ ऑफ दीज वुड अपीयर एज द सैटेलाइट, राइट? इस नॉन कोडिंग पार्ट में यहां तक बात समझ आ रही है? टिल हियर इट्स ऑल क्लियर? ठीक है. इट वॉज सीन, कोडिंग को तो हटा देते हैं. इन द नॉन कोडिंग पार्ट इन सैटेलाइट डीएनए देयर वॉज समथिंग कॉल्ड वेरिएबल नंबर ऑफ टैंडम रिपीट्स. लेट्स सी व्हाट दिस मींस. देखा गया कि नॉन कोडिंग पार्ट में, अभी नॉन कोडिंग पार्ट है, इससे प्रोटीन्स तो बनने नहीं. नॉन कोडिंग पार्ट में सबके डीएनए में there was something called variable number of tandem repeats. Let's see what a tandem repeat is first. Tandem repeat. Tandem repeat is this. This is one tandem. Tandem repeats. Same thing repeating. DNA sequence में ये दो strands हैं. इस strand पे ऐसे है A A A. आगे फिर से A A A. फिर ए ए ए. अभी नॉन कोडिंग पार्ट है तो कैसा भी दिख सकता है. ये रिपीट्स हो रहे हैं. यहां पर ऑब्वियसली सामने टी 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 होगा सबके सामने, है ना? अब मेरे डीएनए में ऐसा है, किसी और के डीएनए में ऐसा भी हो सकता है. ए ए ए, देन जी जी जी, GGG, then TTT. तो ऐसे रिपीट्स लोगों के थे एंड वेरी डिफरेंट फ्रॉम ईच अदर, राइट. वेरिएबल नंबर, अलग अलग नंबर के वेरिएबल डिफरेंट नंबर के टैंडम रिपीट्स, दीज आर टैंडम रिपीट्स. अलग अलग नंबर के टैंडम रिपीट्स थे इन एवरीबडी हैड डिफरेंट टाइप्स और डिफरेंट नंबर्स ऑफ दीज टैंडम रिपीट्स इन वेरियस ह्यूमंस. दिस इज नॉन कोडिंग, तो हो सकता है ऐसा डिफरेंस, बिकॉज इट्स नॉट गोइंग टू इम्पैक्ट, it is not going to impact the coding part. It's not going to lead to any sort of gene form or protein formation. These are not genetic components. These are merely part of genome, not part of gene. तो protein नहीं बनना, तो variation भी है, तो कोई बात नहीं. But this could be used for this comparison. आप 30 लाख पूरा सैटेलाइट डीएनए जिसमें 30 लाख बेस पेयर्स हैं, इसको कंपेयर नहीं कर सकते. Very difficult to compare. But you can check how many tandem repeats are there. If you are able to check this tandem repeats, you will be able to check and do DNA fingerprinting. तो आप सैटेलाइट डीएनए में से आप ये टैंडम रिपीट्स निकाल लो, ये तो कर सकते हैं. दिस इज पॉसिबल. You check how many repeats everyone has, and on the basis of that you will see a pattern, and on the basis of that pattern you will be able to differentiate who was present at the crime scene and who was not. This is the process of DNA fingerprinting. So in DNA fingerprinting, we first take satellite DNA. In that we check for VNTRs. Then we compare VNTRs. तो एक NCERT का डायग्राम दिखाता हूं आपको of DNA fingerprinting. Looks like this, right? I hope this is visible, right? This is, I don't know why it's here. Okay. देखो, let's say ये देखो, this is the number of short tandem repeats or variable number of tandem repeats that was found at the chromosome, that was found in the DNA. कि यार क्रोमोसोम 1 पे इतने टैंडम रिपीट्स मिले, क्रोमोसोम 7 पे इतने टैंडम रिपीट्स मिले, क्रोमोसोम 2 पे इतने टैंडम रिपीट्स मिले, राइट? ऐसा आपको मिला. क्रोमोसोम 7 लुक्स लाइक दिस, नॉन कोडिंग पार्ट में रिपीट्स हो रहे हैं. क्रोमोसोम 7 looks like this, chromosome 2 looks like this, chromosome 16 looks like this, etc, etc. You found this in the crime scene, right? So 3 chromosome, chromosome 7, chromosome 2, chromosome 16, इन 3 chromosomes में इस टाइप के tandem repeats मिले. You put this on, when you isolate these tandem repeats, you will put this on some substance. Which substance I'll tell you. You will put this on this substance, you will see this pattern. You have 2 suspects. You take their DNA and you make their pattern of tandem repeats. Now you can see here that this is the one that was found at the crime scene. आपने यहाँ पे tandem repeats देखे और आपने यहाँ पे tandem repeats वाला DNA एक जगह से रख दिया. This is from crime scene. Then there were 2 suspects. इनके tandem repeats निकाले, इनके tandem repeats निकाले. Now you see this is matching. Now this is easy to compare. You don't have to read anything. The pattern is visible. There is some process through which we can put these tandem repeats like this. वो कैसे होती है वो अभी दिखाऊंगा. But if you are able to do this, then the comparison would become very easy. You just have to see the pattern of these tandem repeats. अब यहाँ पर ज्यादा tandem repeats हैं, यहाँ पर इस आदमी के कम tandem repeats हैं. Right? Everybody has different type of tandem repeats because it's in the non-coding part. In the satellite DNA, it doesn't matter. Right? समझ आ गई बात? Are you basically understanding what we are doing? Screenshot from the NCERT. Take the tandem repeats out, just compare the tandem repeats. Everybody has different. अच्छा. Now think. I'll ask you one last question. अगर आपको समझ आया तो आप इसका सही जवाब दे पाओगे. Can I use this comparison of VNTRs, variable number of tandem repeats? इसका comparison, can I use this for checking if there is a paternity dispute? How does a paternity dispute happen? Also, it's never called a parental dispute. It's always called paternity dispute because there is no doubt as to who is the mother of the child. because child emerges from the mother has carried the child, so mother में कभी doubt नहीं होता. It's only males who have a doubt whether the child is there or not. Mother में तो क्या ही doubt है, she is bearing the child, right? So let's say this is a child born to a mother called M1. Now there is a dispute whether this individual P1, or let's call him F1, whether F1 is the father or F2 is the father. There is a dispute. Can I use VNTRs to solve this dispute? And if yes, how? If yes, how? If no, why? सोच के बताना, जल्दी आंसर मत लिखना. सोच के, yes no नहीं चाहिए, एक्सप्लेनेशन चाहिए. yes because coding, no because non coding, इससे क्या लेना देना है यार? What are you trying to say when you say yes because coding or no because non coding? कोडिंग नॉन कोडिंग से क्या लेना देना है? मुझे एक बात बताओ. लेट्स से माय डीएनए ऑन क्रोमोसोम नंबर 1. चलो अब समझते हैं. क्रोमोसोम 1, मेरा जो क्रोमोसोम 1 है, आई हैव गॉटन लेट्स से दिस फ्रॉम माय फादर, this from my mother. The chromosome that I've gotten from my father has 8 tandem repeats. The chromosome that I've gotten from my mother has 6 tandem repeats. ठीक है, थोड़ी बहुत. अब इसमें कोडिंग नॉन कोडिंग का क्या लेना देना है? I have gotten all my DNA from my parents. There are 8 tandem repeats I have gotten from my father. Maybe my father had 9 tandem repeats. थोड़ा बहुत हो सकता है क्रॉसिंग ओवर हो गया हो. But if you now have to compare the paternity, at least the VNTR pattern that the child will exhibit, child has half DNA from father. In that half of the DNA, the VNTR pattern that the child will exhibit will be very similar to the father or not. It will be very similar, much more similar if let's say he is the real father, much more similar to him than it would be to him. है ना? अब हाउ इज सेक्स ऑफ चिल्ड्रन डिटरमिन्ड, ये आप लोगों को पता होगा. एक्स क्रोमोसोम एंड वाई क्रोमोसोम मींस बॉय, एक्स क्रोमोसोम एंड एक्स क्रोमोसोम मींस गर्ल. है ना? दैट मींस अब देखो, यही मैं चाहता था कि आप थोड़ा सोचो और जल्दी जवाब ना दो. ये होता है इफ दिस इज अ बॉय चाइल्ड, दैट मींस फादर हैज गिवन वाई क्रोमोसोम. वाई क्रोमोसोम तो पूरा पूरा फादर से आया, right? Mother can't give X chromosome. Mothers have this, so the female gamete always carries the X chromosome. Fathers have X and Y chromosome, so the sperm can carry either X or Y. If the boy is a male or the child is a male, father must have given Y chromosome, mother must have given X chromosome. Therefore, there is a boy, right? Ab Y chromosome has come 100% from the father. In this child, in this person, Y chromosome has come 100% from the father. You compare Y chromosome VNTR pattern from F1 and Y chromosome VNTR pattern from F2. It will be very similar to F1 as compared to F2, so you can use it for paternity disputes. अगर मैं इससे लोगों का डीएनए आइडेंटिफाई कर सकता हूं, तो मैं पेरेंटल भी तो कर सकता हूं. आई कैन इवन कंपेयर टू पेरेंट्स, राइट? समझ आ रही है बात? अब repeats will be different from everyone, but where is my DNA coming from? आया कहां से मेरा different? You are saying because tandem varies in all, but where did my DNA come from? थोड़ा बहुत चेंज हुआ होगा क्रॉसिंग ओवर में, पैटर्न तो सिमिलर होगा. My father, let's say, had 10 VNTRs on chromosome 1. I'll be very similar to him. Pattern will be similar. अरे तो only okay. Somebody is writing only in case of male child. Even in female child, you can do this comparison. Do it for multiple chromosomes. Male child mates easier to do. You do it for female also. Female has gotten their entire DNA from their parents. इसका maternal chromosome है, एक father का chromosome है in chromosome number 1, right? एक mother से आया, एक father से आया. इसका यहां पर VNTR pattern check कर लो. Check the VNTR pattern here. Check the VNTR pattern here. This VNTR pattern would be very close to mother. So अब mother की chromosomes की बात कर लेते हैं. Mother का आप एक VNTR pattern निकाल दो. Father का एक VNTR pattern निकाल दो. This pattern अगर आप दोनों का साथ में निकालोगे would be half similar to this, half similar to this. Pattern है ना. Maybe some differences are there. 8 की जगह 4 हैं tandem repeats, but the pattern will be very similar, right? You can do it for all chromosomes. यहां पर आपने पिक्चर शायद ध्यान से नहीं देखी. Look at this. हमने 3 क्रोमोसोम्स का ऐसे पैटर्न निकाला. ऐसे पैटर्न तो आप हर क्रोमोसोम का निकाल सकते हो. ऐसे 23 पेयर्स का आप ऐसा पैटर्न निकाल सकते हो. तो आप इस इंडिविजुअल का निकालोगे. When you take it out for this individual, right? Look at this individual. ये इंडिविजुअल है. इनके क्रोमोसोम 1 पे इतने टैंडम रिपीट्स हैं, क्रोमोसोम 2 पे इतने हैं. तो पैटर्न कैसा बन रहा है? कुछ ऐसा, राइट? दिस हैज कम हियर, दिस हैज कम हियर. क्रोमोसोम 7 पे, दिस इज क्रोमोसोम 7, सॉरी, क्रोमोसोम 2 पे टैंडम रिपीट्स हैं, वो यहां आ रहे हैं, ये यहां आ रहे हैं. अब ये रिपीट्स किस चीज के हैं? ए के रिपीट हैं, जी के रिपीट हैं, सी के हैं या टी के हैं. ये भी तो डिफरेंट होगा, right? Even this would be different. Very similar to the parent it would be, right? थोड़ा बहुत crossing over. And you can make a pattern for all chromosomes. For all 23 chromosomes, you can make a pattern, right? So it's possible here, for example, that these are repeats. The one shown in red are repeats of, let's say, A. These are repeats of G. These are repeats of C. So in this person, on chromosome 11, there are 7 repeats of A. On the second pair of chromosome 11, there are 8 repeats of A. यहां पर 6 repeats of A, 5 repeats of A, 6 repeats of C, 5 repeats of C. Pattern तो आप देख सकते हो ना. Then this pattern would look very similar to their parents' pattern or not? As compared to someone who they are not related to. इन्हें DNA मिला तो सारा parents से ही है. This chromosome has come from the father. Some crossing over, some modification, but the pattern would look very similar. तो आप pattern तो compare कर ही सकते हो ना, या नहीं? It will not be exactly similar. Right? It would not be exactly similar. But half of my DNA has come from my mother. Half of my DNA has come from my father. So half of the pattern will be same. आप pattern देख के समझ जाओगे. If somebody is unrelated, let's say you are unrelated to me. If we compare our DNA patterns, it will be completely different. This is the idea of DNA fingerprinting. So what do you do in DNA fingerprinting? Identification of criminal by use of biotechnology and various other things. We use something called variable number of tandem repeats. It is used to develop DNA bands. देखें ना, हमने ऐसा पैटर्न एक दिख जाता है, ऐसा DNA bands in gel electrophoresis and identify a person. Gel electrophoresis तो हमने देखा ही है पहले क्या होता है. And we identified a person. Can be used in criminal investigation, paternity testing, immigration cases. Criminal investigation तो समझ आ रहा है, paternity testing भी समझ आ रहा है. Now, if it can, how can it be used in immigration cases? बताओ. अब ये सोचो, चाइल्ड स्वैपिंग समझ आ रहा है कि हॉस्पिटल में आप गए, किसी ने चाइल्ड, आप all children that they are born, they are kept in the nursery. Now let's say my child was kept on bed number 1, someone else child was kept on bed number 2. Somebody picked up child from bed number 1 and gave me child from bed number 2, and I think my child has been swapped. So I can check his VNTR pattern or her VNTR pattern with me and the mother of the child's pattern, and it will be matched, right? Disaster victim identification. There are victims in a disaster whose only limbs are found. Let's say सिर्फ उनके limbs मिले, right? From those cells you can extract DNA. Then check with old records. If you have their DNA records, you can identify who this person is, है ना? But how will you check for immigration cases? How will you check for immigration cases? ये बताओ, सोचो थोड़ा सा. अरे एथनिक डीएनए से नहीं होगा, एनवायरमेंट से नहीं होगा. Correct. If there is someone who is trying to immigrate and they say that my parents were citizens of this country, that these two people are my parents, they are citizens of this country, therefore I am citizen of this country. Citizenship में law है ना, हमारे में भी. अब याद करो हमारे में क्या conditions हैं. Conditions for gaining citizenship, conditions for removal of citizenship. In immigration cases, you check again the DNA of parents or grandparents and check if they are lying. Acha. So let's say I am claiming citizenship of India. I say these are two people, they are my parents, and I know that these two parents or these two people are citizens. तो उनके DNA से मेरा DNA check कर दो. If I am actually their child, I will get the citizenship. चलो, one last question to see if you have understood what I am asking here, और आप confused नहीं हुए हो. How much of your DNA comes from your grandmother? बताओ. How much of your DNA comes from grandmother? अरे 99% कौन लिख रहा है यार? कोई लिख रहा है सम अमाउंट. ट्रोल कर रहे हो क्या? अनसर्टन ड्यू टू क्रॉसिंग. अरे भाई चल, क्रॉसिंग ओवर की वीडियो ढंग से नहीं देखी तुमने. लेट्स से फादर इज प्रोड्यूसिंग gametes. During this production of gametes, lots of crossing over happened. Infinite amount of times crossing over happens. Then this goes to the child. How much of this child's DNA comes from the father? How much of the child's DNA comes from the father? Infinite crossing over has happened. How much of the child's DNA comes from father? Still 50%, right? अपने ही आपस में डीएनए में होती है ना यार क्रॉसिंग ओवर, राइट? प्लीज वॉच दैट क्रॉसिंग ओवर वीडियो अगेन. आई विल नॉट एक्सप्लेन इट, और आई विल एक्सप्लेन नेक्स्ट टाइम. बहुत अच्छे से उस वीडियो में बताया है. अपने ही क्रोमोसोम्स में हो रही है ना यार क्रॉसिंग ओवर. इट हैपेंस विद इन योर ओन क्रोमोसोम्स, राइट? फादर हैज क्रोमोसोम पेयर 1, फादर हैज क्रोमोसोम पेयर 1. इनमें आपस में क्रॉसिंग ओवर होती है ना. लेट्स से इट लुक्ड लाइक दिस, it looked like this. After crossing over, it looks something like this. ये ऐसे दिखने लग गए क्रॉसिंग ओवर के बाद. लेट्स से then this will get separated into gametes. One gamete will get this, one gamete will get this, but still is the percentage changing? This gamete will then go to the child, बट है तो ये सारा फादर का ही डीएनए. अपने डीएनए में तो हो रही है क्रॉसिंग ओवर, मदर के डीएनए से थोड़ी क्रॉसिंग ओवर होती है. even if infinite amount of crossing over happens, still father would give 50% DNA only, right? ये समझ आई बात? फादर तो 50% ही देगा ना. अब now let us come to this. How much of your DNA comes from your grandmother? अब I don't think anybody should have been confused in this. Let's say this is your grandmother, this is your grandfather, right? She would have some chromosomes, he would have some chromosomes. Your father would be born here. How much of DNA he would get? He would get one chromosome from, let's say, half DNA from mother, half from father, right? So your father has 50% of DNA from grandmother, 50% from grandfather. अब इसके बाद आप आए हो, when you come, you have 50% of DNA from your father. So you have 50% of DNA, and father's DNA was 50% from grandmother. Father has 50% of grandmother, you have 50% of that 50%. So out of, let's say, 100 units of DNA, your father got 50 units from your grandmother, you got 50 units from your father, so you would get 25 units or not? Please बोल देना ये समझ आ गया है. I'll just write it again. Grandmother, grandfather, 100 units of DNA, 100 units of DNA. He gives 50 and he gives 50 to your father. Father now has 100 units. Out of these 100 units, you have how much? Out of these 100 units, you have 50. So you'll get 50 of this, 50 of this, half of this, half of this. This is then 25 from your grandmother and 25 from your grandfather or not? Now clear? Crossing over might determine this a little bit. Crossing over will change this a little bit, but broadly 25%. This will be impacted by crossing over. This will get impacted by crossing over. समझ आ रहा है how this will get impacted by crossing over? चलो anyway. अब DNA fingerprinting में there is 0.1% different DNA, 99% same. There are some short tandem repeats. Tandem repeats can also look like this. AGAC, 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 ये repeat हुए जा रहा है बार बार. It can look like any repeat. Repeat of AAA, repeat of AGA. Let's say this is repeating. AGA, 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 AGA. ये भी repeat हो सकता है बार बार. Any pattern can be there of repeats. Tandem repeats. Repeat can be any pattern. For example, here AGAC is repeating. Right? Variable number of tandem repeats are there in humans. DNA is classified in two parts: bulk DNA and satellite DNA. Satellite can be classified as both micro and mini satellite depending on the size of it. इतना सा strand है तो micro satellite, इतना सा ही है तो mini satellite. It can be further classified into micro satellite and mini satellite depending on its size. In these, here we look at variable number of tandem repeats and compare. What do? We first isolate the DNA, extract the DNA, we digest the DNA, cut it in various parts, then we put it on gel electrophoresis. तो हम यहां पर देखते हैं, हमने ब्लड सैंपल लिया, हमें डीएनए कंपेयर करना है. लेट्स से मेरे डीएनए को टेस्ट करना है. We took the blood sample, from this we will extract the DNA, then we will cut the DNA. DNA तो बहुत बड़ा मॉलिक्यूल होगा, इसको कट करेंगे, then we will do centrifugation. From that centrifugation, we will identify the satellite DNA. This we will then put on gel electrophoresis. Gel electrophoresis यहां पे है. जेल इलेक्ट्रोफोरेसिस में हम क्या करते हैं, हमें पैटर्न देखना है, तो अगेन वी पुट पॉजिटिव चार्ज हियर, डीएनए इज नेगेटिवली चार्ज्ड, सम विल लीव हियर, कुछ ऐसा ऐसा सा कुछ पैटर्न बनेगा. दिस पैटर्न इज मेड, देन आई हैव टू कंपेयर दिस पैटर्न. अब इस पैटर्न को कंपेयर करने के लिए आई हैव टू ब्लॉट दिस, ट्रांसफर दिस ऑन अ शीट ऑफ नाइट्रोसेल्यूलोज. ये जेल इलेक्ट्रोफोरेसिस की प्लेट है, हमने देखी थी लास्ट टाइम जेल इलेक्ट्रोफोरेसिस. दिस इज अ प्लेट ऑफ जेल इलेक्ट्रोफोरेसिस. I have to transfer this on a sheet of nitrocellulose so that this comparison can happen. Why this transferring has to happen, this also we'll understand. We have to transfer this on a sheet of nitrocellulose. This transfer process is called blotting, and this technique of blotting is called southern blotting. इसका नाम है टेक्निक का southern blotting. Name of this technique is southern blotting, to transfer this from gel electrophoresis to this band. Then we will do something called hybridization with radioactive probe. ये क्या होता है ये कल समझेंगे. Then do subradiography, right, or autoradiography, any of these. Then we will then compare these patterns. तो broadly हम क्या कर रहे हैं, we are taking a blood sample, cutting it into small pieces, then centrifuging, then getting the satellite DNA out. Then for getting VNTRs, what we will do, we will then put it on gel electrophoresis. तो रिपीट के पैटर्न्स, जिसका बड़ा रिपीट पैटर्न होगा वो यहीं रह जाएगा, स्मॉल रिपीट पैटर्न होगा तो ज्यादा आ जाएगा आगे. Then we will get a pattern, but I can't compare on the gel plate because I can't see fragments of DNA on the gel plate. I have to transfer this to a nitrocellulose based paper. This transfer to nitrocellulose based paper is also called blotting. This technique is called southern blotting. Then here I will compare based on radiology. अब इन पे मैं कुछ कलर लगाऊंगा DNA पे, and then I will compare. This is the way of comparing. Ultimately you will see a pattern like this. जैसे यहाँ देख रहे हो आप, ये वही पैटर्न है, the same pattern that we had discussed. So again let's look at the process. ब्लड सैंपल था, ब्लड सैंपल से डीएनए एक्सट्रैक्ट किया, कट किया, सेंट्रीफ्यूज करके यहां डाला, जेल इलेक्ट्रोफोरेसिस कराया. आफ्टर जेल इलेक्ट्रोफोरेसिस आई हैव टू ट्रांसफर टू दिस नाइट्रोसेल्युलोज शीट. फ्रॉम हियर, दिस इज फ्रॉम द एनसीईआरटी बाय द वे, फ्रॉम हियर आई डू समथिंग सो दैट दिस बिकम्स विजिबल, कॉल्ड रेडियोग्राफी. अभी ये पैटर्न ऐसा दिखता है बट इट्स नॉट विजिबल टू द नेकेड आई. इसको विजिबल करने के लिए आई डू समथिंग एंड देन आई कंपेयर इट ऑन दिस. अब ये एक पैटर्न मिल गया. ऐसे और लोगों के टैंडम रिपीट्स के बेसिस पे और लोगों के पैटर्न्स मिल जाएंगे. Somebody's might look like this, somebody's will look very very similar, right? Somebody will look very very similar. Then this would lead to VNTR comparison. This is DNA fingerprinting, use of satellite DNA, comparison of bulk and satellite DNA. Clear? We'll see radiography again. Radiography वाला पार्ट हमने नहीं देखा है अभी, ठीक है? All right. अब what we are going to do now, conditional on you reading these slides, is we are going to look at certain questions. Although आज काफी कुछ करना था मुझे, but ठीक है, that's okay. Maybe one class more for at least biotech, but fine, no worries. Had to get through all of this today, but anyway. We have a test of 7 questions now. ठीक है, 7 questions का हम एक टेस्ट करेंगे. I will tell you the cutoff, or let me tell you the cutoff now. Cutoff is 5 out of 7. Cutoff for this test is 5 out of 7. Same test format. We've already done 7 questions. These are the next 7 questions. Same test format. I'll show you the question. Chat I'll just make private. I'll show you the questions. ठीक है, क्वेश्चंस थोड़ा देखना इंपॉर्टेंट है बीच में, ताकि वी ऑल्सो नो द लेवल ऑफ क्वेश्चंस दैट यूपीएससी आस्क्स. याद रखना कि हमें साइंटिस्ट नहीं बनना है, राइट? अगेन प्राइवेट चैट, 7 questions, show the answer, 1 minute time for each question. Start. Yes, 10 out of 14. Sorry, the cutoff is 10 marks. Sorry about that. All right, next. Sorry. Wait. Don't solve this question. Sorry. Don't solve this question. Sorry about that. I am changing the question. Options make it easy. I am changing the question. I am changing the question. Question is now different. Question is, how many of the above statements are correct? Options are 1, 2, 3, 4. Now solve. ध्यान से पढ़ना, ध्यान से. ठीक है, नेक्स्ट. ओके, क्वेश्चन 4. Might not be visible. This is question 5. For this, I am giving 30 seconds. Last question. This question actually tells people whether they can. This question will actually tell me whether you can apply your mind or not. When you are actually understanding or not, this question will tell that. अब देखते हैं. चलो, फर्स्ट क्वेश्चन, जीएम मस्टर्ड. अब यहां पर एक चीज ध्यान से देखना, हमें पता है इसका आंसर. इफ यू कैन जस्ट एलिमिनेट दिस थर्ड स्टेटमेंट, यू कैन एलिमिनेट थर्ड ऑप्शन एंड गेट दैट इट्स 2 ओनली, है ना. दिल्ली यूनिवर्सिटी ने बनाया तो वी नो, इजी टू सॉल्व. बट लुक एट द फर्स्ट स्टेटमेंट. ध्यान से देखो फर्स्ट स्टेटमेंट को. जीएम मस्टर्ड, सेकंड तो करेक्ट है. GM mustard has the genes of a soil bacterium. right? GM soil bacterium that give the plant property of pest resistance. Pest resistance तो है नहीं इसमें, इसमें तो herb resistance थी, herbicide resistance थी एक्चुअली, है ना? बट लेट से मैं herbicide भूल गया, मेरे को याद ही नहीं रहा कि herbicide है. अब मुझे फर्स्ट स्टेटमेंट कैसे पता चलेगी? थैंकफुली 1 and 2 ऑप्शन नहीं है, बट मान लो 1 and 2 ऑप्शन होता. Look at the option very carefully. What does it say? Genes of a soil bacterium, okay, gives plant property of resistance to wide variety of pests. Why is word wide written here? If they just wanted to write pest resistance, they could have written give the plant property of pest resistance. ये wide variety क्यों लिखा गया है? To make it absolutely wrong. समझ आ रही है ये बात? ये लिखा ही यहां पे इसलिए गया है ताकि ये स्टेटमेंट गलत की जा सके. Wide variety of pests. Look out for words like these. ऐसे वर्ड्स होते हैं, नहीं तो वैरायटी ये लिखने की जरूरत नहीं है ना, है ना? So look out for words like this. Anyway, this question was easy. आगे देखते हैं. RNA interference. अब ये क्वेश्चन मैंने चेंज कर दिया था, इसका आंसर 1, 2 and 4 है. Let's look at all of the options. RNA interference से होगा क्या? DNA से RNA बन रहा है, RNA से प्रोटीन बन रहा है, यहां पर ये प्रोसेस इंटरफेयर कर देगी और RNA से प्रोटीन नहीं बनेगा. It can be used in developing gene silencing therapies. Gene will form RNA, but it will not get expressed. Gene will get silenced, correct. Developing therapies for treatment of cancer. How? There are some cancer genes called oncogenes. Oncogenes will make their RNA. If they form their protein, we will get cancer. हम क्या करेंगे? We will stop this RNA. We will interfere with this RNA. RNA interference we will do. बिल्कुल सही बात है. Developing therapies for treatment of cancer. It last question पहले fourth देखते हैं. It can be used to produce crop plants that are resistant to viral pathogens. अगर they are resistant to nematode of Melodyne incognitia, tobacco में अगर ये हो रहा है तो वो भी हो सकता है. If this can be done, even crop plants can be stopped. This also fine. Let's look at the third statement now. Third statement देखते हैं. It can be used in developing hormone replacement therapies. Hormone replacement. इसको समझते हैं. What is hormone replacement? Hormone replacement means my organs are producing some hormone. Let's say my hormone. What happens is my यार प्लीज चैट बंद कर दो, इट जस्ट डिस्ट्रैक्ट्स मी, प्लीज वेट, है ना? माई लेट्स से देयर इज अ फीमेल एंड शी इज प्रोड्यूसिंग अ हार्मोन कॉल्ड ईस्ट्रोजन. वो ईस्ट्रोजन प्रोड्यूस कर रही है, राइट? व्हेन शी ग्रोज ओल्डर शी विल, okay, I'll stop the chat if you guys want. Right. If this woman is producing estrogen, ठीक है, she is producing estrogen, she ultimately when she reaches the stage of menopause, that is her menstrual period stop, she will have less of this hormone in her body. And now I need to replace this hormone. I need to give her something so that she is still able to produce this hormone. For that, one thing I can do is I can give her some genes, and these genes will code for RNA, and these RNA can be for a particular hormone. ये हो सकता है. But how will I use RNA interference for this? इसके लिए तो मुझे जीन एक्सप्रेस करानी पड़ेगी ना. I will have to have the gene expressed. RNA interference can only stop a gene. It cannot make a gene express itself. समझ आ रही है बात? RNA interference is to stop this process. I can't express this gene, right? It can stop oncogene 100%. It can be used. It can be used. It's not said it is used. It can be used for developing therapies for treatment of cancer. Yes, it might be possible. It occurs in all eukaryotic organisms. ये हमने पढ़ा था. Humans are eukaryotic organisms. I might be able to modify RNA interference in such a way that I can use it to stop the expression of oncogenes. This is not microRNA, by the way. microRNA is completely different. वो हम देख चुके हैं क्या है. This is completely different. है ना? But it can be used. oncogenes के RNA को मैं रोक सकता हूं. यहां तो मुझे कोई जीन एक्टिवेट करानी है. I can't activate a gene by interfering with its RNA. Right? So I can't use this. This I can't do with RNA interference. But I can use crop plants. I can produce crop plants that are resistant to viral pathogens. I was producing a crop plant of tobacco that was resistant to a pathogen called MI. It can be resistant to some other pathogen also. This I can do. अब समझ आया? अब देखो. Now I read one comment here. And now you will tell me why this statement is absolutely wrong. Somebody had written this in the comments. All of you will tell me why this is absolutely wrong. Somebody had said hormones, organs का काम होता है. It is not related to DNA, RNA. Why is this statement absolutely wrong? Why is this statement absolutely wrong? ये बिल्कुल गलत क्यों है ये? And why does this tell me that this person has not revised the first class? आपने भी नहीं किया रिवाइज. Very good. Good. Organs are made of cells. अरे cell is functional unit of life. All functions happen at cellular level. Organ में cell होंगे. This is the organization of life. Cell to tissue to organ to organism. Actually, there is first organ system. To organism. So, cell to tissues in digestive system, to the organ of stomach, to the digestive system as a whole, to the organism which is human. है ना? Cell is structural and functional unit of life. All function happens in cell. So even hormone production will be happening in some cell. One hormone is insulin. We read beta cells. Beta cells were producing insulin. Pancreas was the gland. है ना? ये सारी बात हमने करी है ना यार. If you write statements like this, then it's not okay. That means you are not studying properly. ये गलतियां मत करो. I know मैं डांट रहा हूं, मैं इसीलिए डांट रहा हूं because so that you don't mess this up in the prelims. Ultimately आप यहां पर कर सकते हो गलती, but should not be happening in the prelims. अब देखो, this is a tough question. ठीक है, ऑप्शंस ने इसको आसान बना दिया बिकॉज ये 2019 में आया था. अगर ये अभी आता तो हो सकता है ये 1 only 2 only 3 1 करेक्ट 2 करेक्ट 3 करेक्ट 4 करेक्ट आ जाता. हाउ मेनी स्टेटमेंट्स आर करेक्ट आ जाता. इट्स इंपॉर्टेंट टू अंडरस्टैंड द कॉन्सेप्ट. है ना, कॉन्सेप्ट समझना जरूरी है, रिविजन जरूरी हैं. तभी आई एम ट्राइंग माई लेवल बेस्ट कि आप रटो ना कुछ भी. अभी तक तो कुछ नहीं रटा है हमने, है ना. इसीलिए दिस क्वेश्चन इज डिफिकल्ट. और जो लोग इस क्वेश्चन को आप पढ़ाएंगे वो जैसे यू कैन यूज सम वियर्ड लॉजिक, हार्मोन्स आर नॉट रिलेटेड टू डीएनए तो इसलिए गलत है. ऐसा नहीं है, राइट. यू हैव टू स्टडी दिस इन डिटेल बिकॉज वो पूछ रहे हैं इन डिटेल. दिस इज अ डिफिकल्ट क्वेश्चन टू सॉल्व. आगे दिखाऊंगे और भी क्वेश्चंस डिफिकल्ट हैं। अब आर डीएनए टेक्नोलॉजी, हियर यू शुड हैव नो प्रॉब्लम। यहां आपको कोई प्रॉब्लम नहीं होनी चाहिए थी। हेलो, अब ठीक है? आई थिंक नाउ इट शुड बी फाइन, शुड बी फाइन नाउ। नाउ इज इट ओके? या सो प्रिंसिपल ऑफ कॉन्टिन्यूटी ऑफ डीएनए, दैट इज व्हाई दिस इज परमिसिबल। अब ये इतने आसान क्वेश्चन कब आते थे? 2013 में, ठीक है। अब mycorrhizal biotechnology has been used in rehabilitating degraded sites because mycorrhiza enables the plant to resist droughts and increase adoptive area, yes. Tolerate extremes of pH, yes. Resist disease infestation, yes. Right? समझ आ गई बात? अब नेक्स्ट क्वेश्चन, विद रेफरेंस टू एग्रीकल्चर इन इंडिया, आरएनए इंटरफेरेंस पे आता हूं वहां पे अगर वापस कोई डाउट है तो, बट विल कम टू दिस। विद रेफरेंस टू एग्रीकल्चर इन इंडिया, हाउ कैन टेक्निक ऑफ जीनोम सीक्वेंसिंग ऑफन सीन इन द न्यूज बी यूज्ड इन इमीडिएट फ्यूचर? मतलब मैं पूरा जीनोम निकाल लूं, नाउ आई नो द एंटायर जीनोम, मुझे सब पता है यहाँ पे कहां पर ATGC क्या लिखा है, कहां पर कौन सी जीन्स हैं, व्हाट इज जीन, व्हाट इज नॉन कोडिंग पार्ट, व्हाट इज कोडिंग पार्ट, मुझे सब पता है अब डीएनए के बारे में। नाउ हाउ कैन आई डू दिस? व्हाट कैन आई डू विद दिस? कैन बी यूज्ड टू आइडेंटिफाई जेनेटिक मार्कर्स, जीन मार्कर्स और डिजीज रेजिस्टेंस एंड ड्रॉट टॉलरेंस। मैं डिजीज रेजिस्टेंस की जीन और ड्रॉट टॉलरेंस की जीन आइडेंटिफाई कर सकता हूं, बिल्कुल कर सकता हूं। helps in reducing the time required to develop new varieties of crop plants, बिल्कुल। अगर मुझे पता है that this wheat, I look at its genome and I know that okay, this has disease resistant and very good genes, this is wheat too, this has very good genes of yield, then I can merge them. I don't have to test in the field, I don't have to test in the field, right? It can be used to decipher host pathogen relationships in crops, बिल्कुल। होस्ट पैथोजन रिलेशनशिप्स, अभी वैसे वायरस पढ़ेंगे तो ये लाइन और क्लियर हो जाएगी, बट होस्ट पैथोजन रिलेशनशिप वाज आल्सो बीइंग डिटरमिन्ड इन आरएनए इंटरफेरेंस, राइट? सो ऑल दीज थिंग्स आर पॉसिबल। अगर मुझे पूरा डीएनए पता है, मेरे को ये सारी चीजें पता चल सकती हैं। I can know what is genetic markers I can find for disease resistance and for drought tolerance. I can find what are beneficial genes, so it will help me reduce the time required, and I will also understand host pathogen relationships, है ना? अब फोर्थ स्किप थोड़ी किया था, तो फैक्ट है तो डायरेक्ट स्लाइड से। क्वेश्चन 6. अब this I definitely did not tell you directly. अब दिख रहा है? वैसे क्वेश्चन तो तुम्हें पता ही है, देख तो तुम सकते ही हो. If the voice is clear, why is this problem? Anyway, ball guard 1 and ball guard 2. I did not specifically tell you the name of this technology because I wanted you to do this word breakdown. Protection against bollworms. Protection against bollworms. BT cotton. Answer is B. Ab microsatellite DNA. This question actually came in 2023 prelims. Slightly difficult to solve, I would say. Okay, slightly difficult. Why is it difficult? I can use satellite DNA to check for VNTRs. With VNTRs, I can compare my DNA and my father's DNA. I can compare my father's DNA to his grandfather's DNA, to his grandfather's DNA, to his grandfather's DNA, to his grandfather's DNA, to probably our ancestor, and check this evolutionary relationship. Right? To check this evolutionary relationship. स्टेम सेल से कोई लिंक नहीं है माइक्रोसैटेलाइट डीएनए का, स्टेम सेल्स हम कम पढ़ेंगे. क्लोनल प्रोपोगेशन, अरे रुक जाओ यार, स्कोर बता दें, रुक जाओ. Clonal propagation is something that you might have heard in lot of questions. This option is there. So I otherwise would not have told you clonal propagation, but I will teach you clonal propagation otherwise. ठीक है, वैसे मैं नहीं पढ़ाता, बट 3-4 जगह मैंने ऑप्शंस में ये स्टेटमेंट देख ली है, और हो सकता है क्लोनल प्रोपोगेशन पे क्वेश्चन आ जाए. ऑप्शन एनालिसिस इज इंपॉर्टेंट. लास्ट इज efficacy of drugs by conducting series of drug trials in a population. Obviously no relationship with this because microsatellite DNA is the different DNA, mostly non-coding DNA. यहां पर कोई कहानी है ही नहीं. ठीक है, now let us come to your scores. आप स्कोर बता सकते हो मुझे? Most people I think have gotten more than 11. 11.34 is the most common one. एक क्वेश्चन गलत किया है तो देख लो यार. If you are compared Saurabh is saying this chat is a curse to see. I have to see it all the time, right? I have to see it all the time. आप तो अभी देख रहे हो. देखो, अगर मार्क्स थोड़े बहुत कम हैं, इट डज नॉट मैटर. कॉन्सेप्ट वाइज अंडरस्टैंड करोगे, कॉन्सेप्ट वाइज अंडरस्टैंड करोगे तो बात बनेगी. अगर कॉन्सेप्ट नहीं समझोगे तो बात नहीं बनेगी. साइंस एंड टेक कॉन्सेप्ट से ही समझ आ सकता है और कोई तरीका नहीं है, राइट? इट इज कॉम्प्लेक्स, इट इज कॉम्प्लेक्स. ये क्वेश्चंस अगर मैं आपको पहले दे देता बिफोर टीचिंग यू, तो आपका कितना स्कोर आता और अब आपका कितना स्कोर आया, ये देख लो. ठीक है, सो फार वी हैव अटेम्प्टेड 14 PYQs. देयर आर टोटल 110 PYQs दैट वी हैव टू डू. हमने अभी तक 14 कर लिए हैं. 11 years PYQ we are doing, 11 to 12 years, approximately 10 per year, 115 116 का नंबर है. ठीक है, 14 PYQs हमने कर लिए हैं. नेक्स्ट PYQ टेस्ट विल बी ऑन थर्सडे. थर्सडे को होगा नेक्स्ट PYQ टेस्ट. इसमें आज से जो पढ़ाया है उसी पे क्वेश्चन आएंगे. प्लीज स्टडी वेल फ्रॉम नाउ. फ्रॉम नाउ प्लीज स्टडी वेल. आप कॉन्सेप्ट स्किप करते जाओगे तो आपको समझ नहीं आएगा, राइट? यू नीड टू हैव दीज कॉन्सेप्ट्स इन योर माइंड. कोई रात को भी आपको जगा के उठाए तो आपके मुंह से DNA to RNA to प्रोटीन निकलना चाहिए. ठीक है, एज यू कैन अंडरस्टैंड, आई कि यार प्रद्युम प्लीज स्टॉप स्पैमिंग, राइट? As you can understand, it takes a lot of time for me to explain here. टाइम लगता है, it is difficult to explain, तो हमारा थोड़ा टाइम ज्यादा लग रहा है, right? But it's not a lot for you to understand and study from here. तो एक तो अभी ब्रेक मत लेना, ठीक है, और अभी इसको रिवाइज कर लेना. I will try to go a bit faster now. We might need one extra class for biotech. I think we are one class behind. ठीक है, I think we need one extra class for biotech. वो हमें करनी होगी, ठीक है. अब now I will see you tomorrow at 9 AM. 9 AM कल जो हम पढ़ने वाले हैं, we will do viruses, CRISPR-Cas9, we will do gene therapy. CRISPR के साथ we will also see other gene editing technologies. जो आज की चीज थोड़ी सी रह गई, that was transgenic animals. And we will study main part of vaccination. I will have one extra class for biotech. One extra class will be there for biotech. कल हम ये करेंगे, ठीक है. चलो, all right, see you tomorrow. Bye bye.